देव जी महाराज and परम श्रद्धेय आचार्य बालकृष्ण जी. May I request Professor Luis Felix Notias? Thank you. Professor Tidharat Duyangong, Miss Annie Mary Shoni Podon, Professor Y.K. Gupta sir, Professor Pulok K. Mukherjee sir, Professor Major M.D. Ray, एवं आदरणीय पूज्य साध्वी डॉक्टर देवप्रिया जी. अब आचार्य
सर सर्वप्रथम हम अपने पूज्य गुरुवर एवं संस्थापक परम पूज्य योग ऋषि स्वामी रामदेव जी महाराज तथा परम श्रद्धेय आचार्य बालकृष्ण जी महाराज के पावन चरणों में कोटि कोटि नमन अर्पित करते हैं. आज हम सभी सम्मानित अतिथिगण, सम्मानित सहकर्मियों तथा पतंजलि विश्वविद्यालय बीएनएस के पतंजलि आयुर्वेद कॉलेज के प्रोफेसर एवं प्रिय विद्यार्थियों का हार्दिक स्वागत एवं अभिनंदन करते हैं. साथ ही इसी सभागार में उपस्थित परम पूज्य साध्वी देवप्रिया जी, परम पूज्य स्वामी परमार्थ देव जी, डॉक्टर अनुराग वार्ष्णेय जी, डॉक्टर वेद प्रिया आर्या जी, प्रोफेसर अनिल कुमार जी, प्रोफेसर सीबी धनराज जी, श्री राकेश मित्तल जी, डॉक्टर कुलदीप सिंह जी, पूज्य स्वामी अष्टदेव जी, डॉक्टर जगदीप आर्या जी, प्रोफेसर मयंक अग्रवाल जी, मिस अंशु शर्मा जी, मिस पारुल शर्मा जी, ब्रिगेडियर टीसी मल्होत्रा जी, डॉक्टर एनपी सिंह जी, डॉक्टर अनुपम श्रीवास्तव जी, डॉक्टर राजेश मिश्रा जी का डॉक्टर गिरीश के जी का हार्दिक स्वागत एवं अभिनंदन करते हैं. Now we will proceed with the lamp lighting ceremony.
पुरोहितै यज्ञस्य देवमृत्विजम् होतारं रत्नधातमं स नः पितेव सूनवेग्ने सुपायनो भव स च स्वानस्वस्तये स्वस्ति नो मिमीतामश्विना भग स्वस्ति देव्यदितिरनैर्वणाः स्वस्ति पूषा असुरो दधातु न स्वस्ति द्यावा पृथिवी सुचेतुना स्वस्तये वायुमुपप्रवामहै सोमं स्वस्ति भुवनस्य यस्पतिः बृहस्पतिं सर्वगणं स्वस्तये ॐ स्वस्तिः स्वस्तिः स्वस्तिः।
ओम। अब मैं संकल्प शक्ति के धनी हमारे प्रेरणा स्रोत एवं पद प्रवर्तक परम पूज्य योग ऋषि स्वामी रामदेव जी महाराज से सादर निवेदन करती हूँ कि वे हमारे अतिथिगणों को अपने पावन आशीष वचनों से अनुग्रहित करें।
ठीक, आइए डॉक्टर अनुराग जी बैठिए इधर। सब ओंकार की ध्वनि करेंगे मेरे साथ। ओम भूर्भुवः स्वः तत्सवितुर्वरेण्यं भर्गो देवस्य धीमहि धियो यो नः प्रचोदयात्। ओम शांति शांति शांतिः। आज डॉक्टर अनुराग जी भी इधर ही आ जाओ, कहाँ घूम रहे हैं? बैठे, यहाँ बाईं तरफ सीट खाली पड़ी आपने। हाँ डॉक्टर।
May we now request Dr. Miss Anne-Marie Podon to please come on the dais.
Come my sister. वर्ल्ड के रिनाउंड साइंटिस्ट, ऐसे डिवाइन सॉल्स राइट लेफ्ट साइड में यहाँ पर जिनका भी हमने यहाँ सम्मान किया, और उनके आने से ये पूरा पतंजलि विश्वविद्यालय, पतंजलि रिसर्च फाउंडेशन, हम सब उनको हृदय से प्रणाम अभिनंदन करते हैं। एक बार जोरदार तालियों से सबका अभिनंदन। We are so grateful the whole divine source and the renowned world scientists are here, and we are discussing about herbs, plant to patient। ये मंथन विचार विमर्श यहाँ पर कर रहे हैं कि एक एक जड़ी बूटी, एक एक वनस्पति देखने में एक है। हमारे उपनिषदों की एक ऋचा है, एको वशी सर्वभूत अंतरात्मा, एक ब्रह्म से और उसी ब्रह्म की शक्ति सर्वत्र है। लेकिन उस एकता में अनेकों शक्तियों का, सहस्रों हजारों शक्तियों का संघात। नेचर इज ए अल्टीमेट केमिस्ट। नेचुरल हर्ब्स, जड़ी बूटियां, वनस्पतियां, दीज आर द अल्टीमेट हीलर्स। अभी उसके बारे में जानकारी कम है। तुलसी, अश्वगंधा, शतावर, सफेद मूसरी, ब्राह्मी, शंखपुष्पी, एक एक वनस्पति में 100 से लेकर के 1000 एक्टिव कंपाउंड्स हैं और एक एक कंपाउंड के 100 प्लस 1000 प्लस बेनिफिट्स हैं। जैसे देखने में हम एक इकाई दिखते हैं, हम एक इकाई नहीं है। हमारी एक इस व्यक्तित्व में, इन वन पर्सनैलिटी, we have inside 37 trillion cells, and the activity of cells, 46 chromosomes, 23 genes in each and every cell, and the intra and inter signaling of each and every cell is beyond science. We can not calculate the realistic activities of each and every cell of body. हम कुछ अनुमान लगा सकते हैं, हम कुछ सोच सकते हैं कि कुछ हो रहा है अंदर। हमारे ब्रेन में ही 86 मिलियन और न्यूरॉन्स और उसके साथ में जुड़े हुए सारे 100 ट्रिलियन स्नैप्स, 86 बिलियन तो जो है वो ब्रेन के सेल से, उसके अब 100 मोर देन 100 उनके स्नैप्स, और उनके फिर एक एक सेल के साथ, एक एक सेल हमारे इस ब्रेन के साथ, इस ब्रेन के फिर एक एक सेल के रिसेप्टर्स के साथ, उसके न्यूक्लियस के साथ, उसके माइटोकॉन्ड्रिया के साथ, उसके जीन्स के साथ, उसके अंदर की आंतरिक गतिविधियों के साथ जो रिलेशनशिप है, उसको जब सोचेंगे तो माने एक आश्चर्य से हम भर जाते हैं। तो जैसे हमारी एक पर्सनालिटी देखने में एक लगती है, लेकिन इसके भीतर असंख्य गतिविधियां हो रही हैं। अनलिमिटेड एक्टिविटीज, अनलिमिटेड नॉलेज, अनलिमिटेड इमोशंस इज ऑन गोइंग इन ए नेचुरल वे। लेकिन हम पता नहीं है, दिस ऑल सिस्टम इज वर्किंग इज ऑन ऑन ऑटोनॉमिक्स फिलॉसफी, अपने आप चल रहा है सब कुछ। ऐसे ही एक एक जड़ी बूटी, एक एक वनस्पति में ऐसे ऐसे जीवनीय लाभ हैं। अभी तो कुछ ही देखे हैं हमने, ये चौथी मीटिंग है, कि कोई जड़ी बूटी खाओ, लीवर फंक्शन एकदम रेगुलेट होने लगता है। भूमि आंवला से, पुनर्नवा से, मकोई से, शोनाक से, यहाँ तक कि एरेंड के पत्ते जिनको हम ये समझते हैं तो फालतू में ही हैं। पतंजलि ने पहली बार दुनिया को एरेंड के पत्तों से भी लीवर प्रॉब्लम ठीक की जा सकती है और पपीते के पत्तों से प्लेटलेट्स को अच्छा किया जा सकता है, इसके पूरे साइंटिफिक एविडेंस दिए। और जिनको लोग फेंक दिया करते थे, आज उनको चुन चुन करके एरेंड के पत्ते, पपीते के पत्ते उनको उपयोग में ला रहे हैं। पत्ते का अलग मेडिसिनल कंपाउंड है। द मेडिसिनल कंपाउंड इन लीव्स, इन फ्रूट, इन सीड, इन रूट, एवरीवेयर इज डिफरेंट। और उसको वर्ल्ड में पहली बार मेडिसिनल प्लांट्स और उन प्लांट्स के पंचांग का, मूल, तना, फल फूल पत्तियों का फिंगरप्रिंट भी पहली बार सबसे बड़े रूप में पूरी दुनिया के सामने किसी मेडिसिनल प्लांट्स का फिंगरप्रिंट रखा है तो पतंजलि रिसर्च फाउंडेशन। ये कार्य पूज्य श्रद्धेय आचार्य श्री जी की सन्निधि में डॉक्टर अनुराग जी, डॉक्टर वेद जी और इनके पूरे साइंटिस्टों की टीम, और आज जैसे हमने कहा वर्ल्ड के रिनाउंड साइंटिस्ट हमारे लैब राइट साइड में हैं। एक बार इन सब का अभिनंदन करते हैं कि द जर्नी ऑफ प्लांट टू पेशेंट ये अपने आप में बहुत ही उत्सुकता से भरी हुई है। पूरी दुनिया का ये नया रास्ता है। द होल वर्ल्ड इज लुकिंग नेचुरल मेडिसिन्स, और उस पूरी नैसर्गिकता का जो रास्ता है, बैक टू नेचर, बैक टू कल्चर, बैक टू रूट्स, बैक टू वेदाज, बैक टू आयुर्वेदाज, इसका कहीं से सूत्रपात हो रहा है और पूरी दुनिया को स्वास्थ्य की, शिक्षा की और जीवन पद्धति की नई दिशा कहीं से मिल रही है तो पतंजलि है। और उस पतंजलि में जो एक बड़ी भूमिका होती है, साइंस और टेक्नोलॉजी में हम लोग क्या कर रहे हैं, तो नई रिसर्च, नॉलेज, इनोवेशन, इन्वेंशन, रिसर्च से नया क्रिएशन, पूरी दुनिया इसी के इर्द गिर्द चलती है। और आज हम यहाँ तो अभी उसका उद्घाटन सत्र है। इन अलग अलग जड़ी बूटियों से, जड़ी बूटियों का रस भी प्रयोग किया जाता है, जड़ी बूटियों का चूर्ण प्रयोग किया जाता है, जड़ी बूटियों का क्वाथ कषाय प्रयोग किया जाता है, जड़ी बूटियों का एक्सट्रैक्ट प्रयोग किया जाता है। पूज्य आचार्य जी मेरे से 20 साल पहले बात किया करते थे कि हम फाइटो मेडिसिन तैयार करेंगे, तो हम आचार्य जी से पूछते थे कि ये फाइटो मेडिसिन क्या होती है। बोले ये जो है एक एक मेडिसिन के अंदर कुछ एक्टिव कंपाउंड्स होते हैं और उसको एक कंपाउंड को अलग करके एक सिंगल मॉलिक्यूल के लेवल से लेकर के, सिंगल मेडिसिन से लेकर के, फिर उसके जो मल्टीपल जो उसके केमिकल्स हैं, मेडिसिनल कंपाउंड्स हैं, उनको हम एक साथ कैसे प्रयोग कर सकते हैं, एक मेडिसिन में से एक विशेष उसके औषधीय गुण का कैसे संचय करके उसको उपयोग में ला सकते हैं। तो जो पहले 20-30 साल पहले सोचा करते थे, आज उसको पूरी दुनिया में सबसे पहले जड़ी बूटियों पर आधारित औषधीय शक्ति से क्या क्या हो सकता है। लीवर किडनी प्रॉब्लम्स को रिवर्स किया जा सकता है, पेनक्रियाज के डिजनरेशन को, बीटा सेल्स को डिजनरेट होंगे उनको रिजुनेट किया जा सकता है, हार्ट ब्लॉकेज को रिवर्स किया जा सकता है। ऐसे ही वात पित्त कफ त्रिदोषों को अर्थात अर्थराइटिस को, ब्रोंकाइटिस को और तमाम तरह के ब्रेन नर्वस सिस्टम से रिलेटेड प्रॉब्लम्स को, माने यदि साइंटिफिक लैंग्वेज में बोलें तो किसी भी प्रकार के ब्रेन की डिजनरेशन से, न्यूरॉन्स की डिजनरेशन से लेकर के, नेफ्रॉन्स की डिजनरेशन से लेकर के, मैकुलर डिजनरेशन से लेकर के, माने पूरे बॉडी के डिजनरेट को रिजुविनेट किया जा सकता है। ये पहली बार पतंजलि ने करके दिखाया। वी कैन रिजुविनेट लीवर, किडनी, हार्ट, लंग, ब्रेन एंड द होल सिस्टम। ये बड़ी उपलब्धि है। और टारगेटेड मेडिसिन के साथ साथ इंटीग्रेटेड मेडिसिन, प्लांट बेस्ड मेडिसिन से किया जा सकता है, ये भी पतंजलि ने करके दिखाया। सो वी आर प्रोवाइडिंग हियर टारगेटेड मेडिसिन, इंटीग्रेटेड मेडिसिन, और वी कैन रिजुविनेट द डिफरेंट ऑर्गन्स। और इसीलिए जब कोई पहली बार कोई चीज होती है तो उसके ऊपर आरोप प्रत्यारोप बहुत लगते हैं, हम उसकी परवाह भी नहीं करते। लेकिन नई दिशा ऐसे ही निकलती है। और लास्ट एक बात कह करके, इस धरती पर पूज्य आचार्य बागशन जी का और स्वामी रामदेव का जन्म और पतंजलि का ये संगठन साधारण काम को करने के लिए नहीं है, असाधारण कार्यों के लिए। in health, in education, in research, in agriculture, in industry, everywhere. We are doing the incredible work. और पूरी दुनिया पतंजलि को फॉलो कर रही है अलग अलग क्षेत्रों में। ऐसे इस संस्थान में आज विश्व के यहाँ पर गणमान्य साइंटिस्ट हैं। हमारा तो काम है सबको आशीर्वाद देना, उत्साह देना। अलग अलग यहाँ प्रेजेंटेशन होंगे। और अभी तक तो हम बगल में झोला डालते थे, आज अनुराग वार्ष्णेय जी ने हमारे गले में झोला डाल दिया। थैंक यू बहुत। मैंने कहा ये इतना लंबा चौड़ा पहचान पत्र तो पहली बार देखा। बोले इसमें बहुत माल पानी भी रखा जा सकता है। तो ये अच्छा नई नई क्रिएशन भी करते रहते हैं। हमारा डॉक्टर अनुराग वार्ष्णेय बहुत फनी भी है, वो लाफिंग और स्माइलिंग साइंटिस्ट है। अब जलेबी वाला कहना बंद कर दिया। बहुत लोगों ने इनके लिए जब आए तो जलेबी लाना शुरू कर दिया। तो मैंने कहा भाई ऐसा नहीं है, हमारा साइंटिस्ट अब पतला हो गया है। तो हम एक बार सभी साइंटिस्टों का और पूज्य आचार्य श्री जी के संकल्प का और सारे साइंटिस्टों के पुरुषार्थ का 7000 से ज़्यादा रिसर्च प्रोटोकॉल और लगभग 500 रिसर्च पेपर और अभी 500 की और तैयारी हो रही है। और ये सब कार्य खाली हमारे साइंटिस्ट ही नहीं कर रहे, यहाँ के BMS, BNYS और PhD और अलग अलग प्रकार के जो है शोध अनुसंधान के छात्र, वो भी इन रिसर्च प्रोग्राम्स में सम्मिलित हो रहे हैं। आप सबका भी एक बार बहुत बहुत अभिनंदन है। तो आप सबको ये जो रिसर्च का एक्सपोजर है, बहुत से इंस्टीट्यूट्स में तो पूरी पढ़ाई होली साहब, BMS कर लिया, BNYS कर लिया, PhD भी कर लिया, कोई डीलीट भी कर लिया, अलग अलग प्रकार के और बहुत सारे काम कर लिए। पतंजलि में जो बालक 1 साल रह लेता है, वो धरती पर जितने भी रोग और रोगी उन सबको देख ही नहीं लेता, उनका उपचार कैसे किया जाता है ये एक्सपीरियंस कर लेता है। और आप अपने जीवन में थोड़ा प्रयत्न करो तो कम से कम 100 रिसर्च पेपर लिख सकते हो। इतनी कैपेबिलिटी आपको पतंजलि में रह करके आ जाती है। ये डॉक्टर अनुराग कई बार कहता है कि कोई साइंटिस्ट जीवन में एक मेडिसिन को रिसर्च कर ले या इनोवेशन कर ले तो उसको लगता है मैंने ज़िंदगी में बहुत बड़ा काम कर दिया। यहाँ तो 100 से ज़्यादा बड़ी बीमारियों के लिए ऐसी औषधियाँ जो कि मॉडर्न मेडिसिन में भी जो पॉसिबल नहीं था, वो इम्पॉसिबल कार्य यहाँ पर पॉसिबल हुआ है। इससे बड़ी और उपलब्धि हो क्या सकती है। तो बहुत बहुत धन्यवाद सभी को और पूज्य आचार्य श्री को और पूरी टीम को प्रणाम।
परम पूज्य महाराज जी, आपके प्रेरणादायक मार्गदर्शन के लिए हम आपका हृदय से धन्यवाद और सादर नमन करते हैं। अब मैं आयुर्वेद शिरोमणि परम श्रद्धेय आचार्य बालकृष्ण जी से सादर निवेदन करती हूँ कि वे मंच पर स्वागत उद्बोधन से हम सभी का मार्गदर्शन प्रदान करें।
चलो जी, आचार्य जी के लिए बहुत बहुत अभिनंदन।
भारत माता की वंदे। आज पतंजलि का यह पवित्र परिसर परम श्रद्धेय पूज्य स्वामी जी महाराज की पावन उपस्थिति, उनकी प्रेरणा और हमारे इस मंच पर और मेरे सम्मुख भी विश्व के अनेक देशों से पधारे हुए हमारे वैज्ञानिक विद्वज्जन आदरणीय डॉक्टर प्रोफेसर डोमेनिको विटोरियो डेल्फिनो जी, आप पेरूजिया विश्वविद्यालय, पेरूजिया यूनिवर्सिटी इटली से यहाँ पर पधारे हैं। ये प्रेसिडेंट हैं इंटरनेशनल सोसाइटी फॉर इथनोफार्माकोलॉजी ISE के, और इथनोफार्माकोलॉजी के ऊपर आपको दुनिया जानती है और पतंजलि के प्रति और श्रद्धेय स्वामी जी के कार्यों के प्रति आपकी बहुत ही एक प्रेम और सौहार्द है। तो आपका बहुत बहुत अभिनंदन प्रोफेसर डोमेनिको जी को, बहुत ज़ोरदार एक बार आप करतल ध्वनि से स्वागत करें। और उनके साथ में बैठा हुआ ये जवान सा छोरा हमारा, कल हमारी बड़ी दोस्ती हो गई इसके साथ में। और ये डॉक्टर प्रोफेसर लुई फेलिक्स नाटियस, ये इनकी माता जी भी बैठी हैं इधर। ये माता बहुत अच्छी फोटोग्राफर है, तो मुझे दो तरह से अच्छा लगा। एक तो अपनी माँ को लेकर के घूम रहे भाई साहब, और दूसरा ये माँ बहुत ही अच्छी हैं। तो ये कोट द अजूर यूनिवर्सिटी फ्रांस से ये हैं, तो इनको बहुत ही हमारी शुभकामनाएं हैं। ये एआई पर भी काम करते हैं और अलग अलग क्षेत्रों में ये काम करते हैं। और इसके साथ में यहाँ पर हैं आदरणीय डॉक्टर तिड़ारेट टुआंगयोट। ये बहन जो है हमारी डुआंगयोट जी, ये माइफा लुआंग यूनिवर्सिटी थाईलैंड से हैं, तो इनको बहुत बहुत हमारी शुभकामनाएं हैं। ये लुई फेलिक्स जी भी बहुत सारा काम करती हैं प्लांट्स को लेकर के, बाकी चीज़ों को लेकर के यहाँ पर पधारी हैं। तो हमारे साथ साथ में यहाँ पर प्रोफेसर रूडोल्फ बाउर जी यहाँ पर हैं, ग्राज यूनिवर्सिटी ऑस्ट्रेलिया से। तो ये भी इथनोफार्माकोलॉजी के बहुत रिनाउंड साइंटिस्ट और प्रोफेसर हैं। तो प्रोफेसर रूडोल्फ बाउर को भी हमारी बहुत बहुत शुभकामनाएं हैं। और उनकी मुस्कुराहट भी बड़ी प्यारी है, जितना मैं बोल रहा हूँ सब समझ में आ रही है उनको, ऐसा मत सोच लेना नहीं समझ रहे हैं। साथ में हैं प्रोफेसर एन मेरी शॉनी पोर्डान जी, तो इनको भी हमारी बहुत शुभकामनाएं। ये सीनियर पब्लिशर हैं फार्माकोलॉजी एल्सवियर नीदरलैंड से। जो एल्सवियर का जो फार्माकोलॉजी वाला जो न्यूज़ जर्नल छपता है, उनकी ये चीफ एडिटर हैं जहाँ तक मुझे पता है। तो मेरी बहुत बहुत शुभकामनाएं हैं इनको। और हमारे इस सब कार्य के कर्ता धर्ता हमारे प्रोफेसर पुलक के मुखर्जी जी, तो ये बहुत जुड़े हुए हैं सोसाइटी फॉर इथनोफार्माकोलॉजी SFE, ये जादवपुर यूनिवर्सिटी कोलकाता से हैं। तो इनके ही प्रयास से यह आयोजन यहाँ पर हो रहा है। और हमारे सुपरिचित पतंजलि, जिनको बहुत अच्छी तरह से पहचानता है और वो पतंजलि की गतिविधियों को पहचानते हैं, प्रोफेसर YK गुप्ता जी, जो एम्स कल्याणी भारत में अभी आप कार्य करते हैं। और हमारे बीच में हैं प्रोफेसर हमारे डॉक्टर MD रे, ये भी एम्स नई दिल्ली से हैं। और प्रोफेसर साहब हमारे क्लिनिकल रिसर्च में बड़ा योगदान दे रहे हैं। और आपका बहुत बड़ा पुरुषार्थ है कि हम लोग अलग अलग तरह के रिसर्च के कार्यों में बहुत कुछ कर पा रहे हैं। और साथ में इस मंच पर हमारे डॉक्टर अनुराग जी और डॉक्टर वेद प्रिया जी, दोनों को भी हमारी शुभकामनाएं। बात यह है कि इथनोफार्माकोलॉजी को लेकर के इंडिया में एक बड़ा इंटरनेशनल कॉन्फ्रेंस हो रहा है नाईपर चंडीगढ़ में। कहानी शुरू होती है यहाँ से। तो इथनोफार्माकोलॉजी की जब बात हुई कि नाईपर में हो रहा है, और हमने डॉक्टर मुखर्जी जी को, क्योंकि हमसे पहले से ही जुड़े हुए हैं, हमने कहा कि आपके पास इंटरनेशनल बड़े बड़े साइंटिस्ट आ रहे हैं, रिसर्चर आ रहे हैं, प्रोफेसर्स आ रहे हैं, तो उनको वाया पतंजलि करके कार्यक्रम तय किया जाए। तो डॉक्टर पुलकित जी ने कहा कि ये तो बहुत अच्छा रहेगा, क्योंकि इथनोफार्माकोलॉजी के ऊपर जितना पतंजलि में प्रैक्टिकली ग्राउंड पर जो रिसर्च हो रहा है वर्ल्ड में कहीं नहीं, और वहाँ के वर्ल्ड के साइंटिस्ट को पतंजलि ज़रूर दिखना चाहिए, इसीलिए यह आयोजन संभव हो पाया। अन्यथा इतने सारे साइंटिस्टों को एक जगह लेकर के आना वर्षों की योजना होती है, और उसके लिए श्रम के साथ में अर्थ भी बहुत लगता है। तो ये मुखर्जी जी के प्रयास से हमें सहजता से प्राप्त हुआ। तो इथनोफार्माकोलॉजी बियॉन्ड टाइम्स के ऊपर हमने कहा हम बात करेंगे, क्योंकि पतंजलि अभी प्लांट्स की हिस्ट्री के ऊपर, मेडिसिन सिस्टम की हिस्ट्री के ऊपर काफ़ी कुछ काम कर रहा है। जब मानव सभ्यता की बात आती है, इतिहास की बात आती है, तो हम लोग कहीं ना कहीं अपने आप को 2-4-5 हज़ार वर्षों में सिमट करके रह जाते हैं। तो जब हम 2-4-5 हज़ार वर्ष में सिमटते हैं तो सारी पैथियाँ, पद्धतियाँ, सारी समझ जो हमारी प्राचीन इतिहास, मान्यताएं उतने ही वर्षों में सिमट करके रह जाती हैं। परंतु पतंजलि जो काम कर रहा है वहाँ पर हम मनुष्य सभ्यता को लगभग 28 लाख वर्ष पीछे तक हम ले जा चुके हैं। इसीलिए हमने कहा कि जब बियॉन्ड टाइम्स की बात होगी, इथनोफार्माकोलॉजी की बात होगी, तो पतंजलि बहुत महत्वपूर्ण भूमिका निभाएगा। कल इसके लिए हम बड़ी चर्चा करेंगे और डॉक्टर रश्मि जी बड़ा काम कर रही हैं। मैंने उनको कह दिया था पूरी तैयारी कर लें, तो कल आप सबको बहुत विशेष रूप से पतंजलि के माध्यम से हम क्या कर रहे हैं उसको सुनने और जानने को मिलेगा। मैं तो आज सोच रहा था जब इथनोफार्माकोलॉजी की बात है तो हम जब नॉर्मली जब आप पढ़ते हैं ना स्कूल कॉलेज में, कहा जाता है कि मनुष्य को ह्यूमन इज़ सोशल एनिमल। तो मनुष्य जब सामाजिक जानवर हो तो जो सोच जो है ना वो देवत्व वाली नहीं हो सकती है। तो हमारी संस्कृति कहती है अहम ब्रह्मास्मि तत्त्वमसि, या हम कहते हैं कि हम ईश्वर के प्रतिनिधि हैं, ईश्वर के पुत्र हैं या ईश्वर के रूप हैं। उस संस्कृति से ही उसके सही स्वरूप को जाना जा सकता है। तो यहाँ से जो जानने की बात है, समझने की बात है, हम उसको दुनिया में पहुँचाने का प्रयास करेंगे। दूसरा हम कहते हैं कि मनुष्य तो बंदर की प्रजातियों से बढ़ कर के फैल कर के आया। तो जानवर हैं और बंदर हैं, तो हमारी सोच पुरानी, वो जब दिमाग के अंदर ऐसी चीज़ें बैठ जाती हैं कि पहले तो हम ऐसे मूर्ख रहे होंगे, फिर हम लोगों को बता दिया गया कि ये पाषाण युग था, ये लौह युग था, ये धातुओं का युग था। जब इस तरह की अवधारणाएं, शिक्षाएं, इतिहास हमको बताया जाता है, सिखाया जाता है, समझाया जाता है, पढ़ाया जाता है, कहीं ना कहीं हमारा मन, हमारा मस्तिष्क, हमारा दिमाग इस तरह के सोच और विचार और भावनाओं से ही चलने लगता है। तो उन भावनाओं से परे भी कोई भावना है क्या? उन सिद्धांतों से परे भी कोई सिद्धांत है क्या? क्या इनसे परे भी कोई मान्यता या कोई प्रमाण अवेलेबल है क्या? यदि उन प्रमाणों को यदि हम दुनिया के सामने रखते हैं तो दुनिया निश्चित रूप से उन प्रमाणों को लेकर के विचार करेगी, सोचेगी और उस क्षेत्र में काम करेगी। इस भाव से ये इथनोफार्माकोलॉजी बियॉन्ड टाइम्स की हमने परिकल्पना की है। यह एक बहुत बड़े सोच के साथ में, बहुत बड़ी दृष्टि के साथ में आने वाले भविष्य के लिए नए कहीं ना कहीं हम नए सृजन और नए शोध और नए सोच को पैदा करने के उद्देश्य से इसकी परिकल्पना की गई है। तो यह आयोजना एक बहुत बड़े लक्ष्य को लेकर के तो हमारे मन में था कि आप सब हमारे प्राध्यापक गण, हमारे स्टूडेंट्स विशेष कर करके जो मेडिकल प्रोफेशन में जो बीएमएस कर रहे हैं, बीएनवाईएस कर रहे हैं, उन्हीं बच्चों को हमने कहा कि आपको इसको जानना, सुनना और समझना ज़रूरी है। एक एक साइंटिस्ट को सुनने के लिए, समझने के लिए आपको बड़ा मुश्किल होता है। इन लोगों से आप सब मिल भी पाएँ तो यहाँ श्रद्धेय स्वामी जी की कृपा है, उनका आशीर्वाद है। यद्यपि थोड़ा सा टाइट हो रहा बैठने में, श्रद्धेय स्वामी जी ने कहा कि टाइट ज़्यादा भरे से लगोगे, तो भरे से लग रहे हैं महाराज जी। यहाँ देखो आप, ये इधर कुर्सियाँ पे डलवानी पड़ी, डलवा दी गई है, तो सब आनंद से बैठ करके इसका आनंद ले लो। और ये मुझे कहते हुए खुशी है कि लगभग मैं पहले ही बता देता हूँ कि 2 दिन के बाद में जितने भी यहाँ पर साइंटिस्ट हैं, अभी तो ठीक है हमारी बात को ये कुछ समझ रहे हैं हमारे वाले, बाकी कम समझेंगे, बाद में ये समझा देंगे। मैं इतना ज़रूर कह सकता हूँ कि जिस भाव को लेकर के यहाँ से आए थे उस भाव को लेकर के यहाँ से नहीं जाएंगे, इनके भाव बदले हुए पाएंगे। ये सामर्थ्य है पतंजलि का, ये सामर्थ्य है श्रद्धेय स्वामी जी का, और उसके लिए सामर्थ्यशाली कार्य का। तो हमारी टीम ने, हमारे वैज्ञानिकों ने, हमारे साइंटिस्टों ने जो काम किया है वो अद्भुत है, वो विश्वस्तरीय है, विश्व के जनमानस को हमारी ओर खींच लाने में सामर्थ्य रखता है। तो सबको मेरी बहुत बहुत शुभकामनाएं, सबको बहुत बहुत बधाइयाँ।
आपके अमूल मार्गदर्शन और उत्साहपूर्ण आशीर्वचनों के लिए परम श्रद्धेय आचार्य जी का हम हृदय से आभार व्यक्त करते हैं तथा आपको सादर नमन करते हैं।
Now we would like to felicitate the companions accompanying with our honorable guests on the stage. First of all, I would like to call on the dais Mrs. Christina Boyle, Mrs. Anisha Ray, Mrs. Rita Scaglia, Mrs. Pulok, Mrs. Mukherjee, सुनो शुरू करना। अब हम एक छोटा डॉक्यूमेंट से पेश करना चाह रहे हैं। I would also like to invite Professor Sanjay Jatak on the dais for felicitation by Maharaj Ji and Param Pujya Acharya Ji.
थैंक यू। योग और आयुर्वेद के माध्यम से मानव के दुखों को दूर करने के एक महान और दिव्य संकल्प के साथ परम पूज्य स्वामी रामदेव जी महाराज एवं परम श्रद्धेय आचार्य बालकृष्ण जी निरंतर पूर्ण समर्पण, तपस्या और अटूट परिश्रम के साथ कार्य करते रहे हैं। उनके दूरदर्शी नेतृत्व, प्रेरणादायक मार्गदर्शन और अथाह परिश्रम के कारण आज योग आयुर्वेद को विश्व भर में करोड़ों लोग श्रद्धा और विश्वास के साथ अपना रहे हैं। इसी सोच के साथ वर्ष 2017 में पतंजलि रिसर्च फाउंडेशन की स्थापना की गई। इसका उद्देश्य प्राचीन ज्ञान और आधुनिक विज्ञान और तकनीकी से जोड़कर आयुर्वेद को पूरे विश्व तक पहुंचाना है। आइए इस आधुनिक अनुसंधान संस्थान और इसके उद्देश्य के बारे में एक छोटी डॉक्यूमेंट्री जर्नी ऑफ पतंजलि रिसर्च फाउंडेशन एंड इंट्रोडक्शन के माध्यम से जानते हैं।
Under the visionary leadership of Param Pujya Swami Ramdev Ji Maharaj and Param Shraddheya Acharya Balkrishna Ji, the Patanjali Research Foundation was established in the year 2017. The goal of this organization is to connect our ancient wisdom and knowledge with modern science and technology and convey this information globally. Now, let us learn more about this advanced research institute and its mission through a short documentary titled Journey of Patanjali Research Foundation and Introduction. I would request the guests to go down. ठीक है.
आरोग्यम परमं भाग्यम, स्वास्थ्यम सर्वार्थ साधनम। स्वास्थ्य ही सबसे बड़ा धन है। यही वह आधार है जिससे संपूर्ण जीवन का निर्माण होता है। इसके अभाव में मनुष्य के सभी प्रयत्न अधूरे रह जाते हैं। हजारों वर्ष पहले जब विश्व की अनेक सभ्यताएं अस्तित्व की लड़ाई लड़ रही थीं, तब एक देश था भारत, जो वसुधैव कुटुंबकम की भावना को लेकर आगे बढ़ रहा था, जहां स्वस्थ जीवन का अर्थ रोग मुक्त शरीर ही नहीं बल्कि जीवन का पूर्ण संतुलन था। यह जीवन को समझने, जीने और संतुलित रखने का विज्ञान था। सनातन चिकित्सा पद्धति आयुर्वेद मात्र एक चिकित्सा पद्धति नहीं थी। इसने मानव को सिखाया कि स्वास्थ्य शरीर, मन, इंद्रियों और आत्मा का सामंजस्य है। महर्षि चरक, सुश्रुत और वाग्भट आदि महान ऋषियों ने विभिन्न प्रयोग,
चलो इसको बंद करो
और अनुभव के आधार
फॉर यू
पर
द इंग्लिश वर्जन ऑफ दिस डॉक्यूमेंट्री। सो वी आर गोइंग, अभी नीचे आगे का प्रोग्राम शुरू करते हैं। इसको इंग्लिश में करना चाहिए था। ये तो जानते हैं अपने वाले, तो जिनको सुनाना चाहिए उनके लिए है। तो जब उनको समझ में नहीं आ रहा तो फिर जो है उसको चलाने का कोई फायदा नहीं। थैंक यू।
बहुत बहुत धन्यवाद। अब आगे के प्रोग्राम को हम प्रस्तुत करते हुए I would request all the dignitaries to please go down the stage and have your respective seats. Thank you very much for being on the stage. इसके साथ ही हम एक छोटी सी प्रस्तुति प्रेजेंट करेंगे। That is from Mr. चंद्र मोहन जी. It is on the theme of P2P4. उन्होंने ये स्वतः बनाया है। It's a self-made song from Mr. चंद्र मोहन जी. And he will be singing this song now.
नहीं चला कौन सा वाला हो है माइक आप लोग ये वाला माइक बंद कर दो। ये माइक बंद कर दो। ऑन म्यूजिक ऑन।
जनजातीय अनुभव ऋषियों की दृष्टि औषधि सूत्रों में जीवन की सृष्टि, धरती की गहराई जड़ी बूटी का सार, वनस्पति में निहित उपचार अपार। लोक ज्ञान अब सीमित ना रहे, वैज्ञानिक मानदंडों से बढ़े, प्रकृति से उपचार, प्रयोग से विश्वास, ज्ञान मानव का साक्षात आधार, युगों से प्रवाहित ये अमर ज्ञान, कॉन्स्टिट्यूशन मानव कल्याण, कॉन्स्टिट्यूशन मानव कल्याण। फाइटोकेमिस्ट्री पर सूक्ष्म विचार, बायो एक्टिव तत्वों का विस्तार, क्लिनिकल परीक्षण सुरक्षा मान, एविडेंस बेस्ड बने उपचार विधान, लैब से लेकर क्लिनिकल केयर, ज्ञान बने मानव सेवा का आधार, प्रकृति से उपचार, शोध से प्रमाण, परंपरा संग आधुनिक विज्ञान, युगों से प्रवाहित ये अमर ज्ञान, कॉन्स्टिट्यूशन मानव कल्याण, कॉन्स्टिट्यूशन मानव कल्याण। आयुर्वेद आधुनिक प्रणाली, एकीकृत हो बने नभ कहानी, विश्व मंच पर गूंजे संदेश, स्वस्थ भविष्य का होवे प्रवेश, नव चेतना का स्वर जागे, युगों का विज्ञान फिर हो आगे, जो था प्राचीन आज भी सत्य, समय बदले पर ज्ञान अमर तत्व, प्रकृति से जीवन सेवा का पथ, वो शोध सशक्त तो मानव हो स्वस्थ, कॉन्स्टिट्यूशन यही अभियान, एथनोफार्माकोलॉजी युगों का सम्मान, वो स्वस्थ शरीर तो बने स्वस्थ समाज, कॉन्स्टिट्यूशन यही अभियान, मानव कल्याण यही अभियान। ओम प्रणाम।
मैं चंद मोहन जी और उनकी टीम को इतने सुंदर गायन के लिए बहुत बहुत धन्यवाद करती हूँ। और मेरा आपसे हृदय कृतल ध्वनि से एक बार पुनः उनका अभिनंदन करें। अब मैं इस अंतर्राष्ट्रीय सम्मेलन के विशिष्ट अतिथि आदरणीय प्रोफेसर वाई के गुप्ता सर को मंच पर पधारकर सेटिंग द टोन एथनोफार्माकोलॉजी एंड पतंजलि रिसर्च फाउंडेशन विषय पर विचार प्रस्तुत करने के लिए आमंत्रित करती हूँ। प्रोफेसर वाई के गुप्ता सर अंतर्राष्ट्रीय ख्याति प्राप्त क्लिनिकल फार्माकोलॉजिस्ट, प्रख्यात शैक्षणिक नेतृकर्ता, जिन्होंने 40 से अधिक वर्षों तक न्यूरोफार्माकोलॉजी, क्लिनिकल फार्माकोलॉजी तथा टॉक्सिकोलॉजी का नेतृत्व किया है। आप ऑल इंडिया इंस्टीट्यूट ऑफ मेडिकल साइंस एम्स न्यू दिल्ली, एम्स जम्मू, एम्स भोपाल, इंडियन इंस्टीट्यूट ऑफ टॉक्सिकोलॉजी लखनऊ में डिपार्टमेंट डीन एंड डायरेक्टर के रूप में कार्यरत हैं।
May I now respectfully request Professor Y.K. Gupta sir to please initiate his lecture. Thank you.
परम श्रद्धेय श्री रामदेव जी, परम पूजनीय बालकृष्ण जी और यहाँ पर आए हुए several professors, dignitaries, scientists from across the globe, faculty and students here and the media people here. लोग कहते हैं कि दुनिया में हिंदुस्तान के दो लोग बड़े फेमस हैं, एक रामदेव जी, एक बालकृष्ण जी। रामदेव जी इसलिए फेमस हैं कि इन्होंने अपने मेहनत से दुनिया को दिखा दिया कि योग से हम बढ़िया से बढ़िया इलाज कर सकते हैं कठिन से कठिन बीमारी का, और सस्ते में, सुंदर, टिकाऊ। और हर दुनिया के हर देश में, हर प्रांत में, हर नागरिक को उनका नाम पता है और लोग सम्मान करते हैं। बालकृष्ण जी ने बता दिया कि दुनिया में, हिंदुस्तान में कोई भी जड़ी बूटी ऐसी नहीं है, देयर इज नो हर्ब which does not have a medicinal property. Only thing is you have to discover and use it properly and scientifically. When there are two people like this who are respected, जिनका सम्मान करते हैं, आज जब उन्होंने हमारा सम्मान किया, यहाँ पे बैठे लोगों का सम्मान किया, तो सोचिए हम कितने बड़े बन गए। I think we are Indian, we are proud of such personalities who have raised the standards, the bar of Indian science, Indian ethos, Indian culture, Indian tradition globally. That is what something we are proud of you both, और हमें इस पे हमेशा गर्व रहेगा। मैं हमारे इन लोगों ने पता नहीं कैसी मेहनत करके एक ऐसे आदमी को चुन लिया जिनका नाम है अनुराग वार्ष्णेय, जो पहले बड़े जलेबी के फेमस होते थे। ये जलेबी हम जब भी आते थे तो हम इसीलिए आते थे कि अनुराग हमको जलेबी खिलाएंगे, लेकिन उन्होंने कुछ दिनों से जलेबी पूछना बंद कर दिया। आदेश, स्वामी जी ने आदेश किया कि अब जलेबी मत खाओ, तुम भी मत खिलाओ और डॉक्टर साहब को भी जलेबी मत खिलाओ, लेकिन फिर भी हमको प्यार से खिला देते हैं। ये प्यार ही है जो जोड़ता है एक दूसरे को। दिस इज लव, अफेक्शन एंड फेथ which connects the different pathies, which connects allopathy, which connects Ayurveda, which connects yoga, which connects all other Indian pathies together. Because what is objective is to treat, to maintain the holistic health of the humanity. चाहे किसी भी तरह से करें। And you might have seen here in the stage, I am wearing coat, pant and suit and tie. On the other hand, there is a person just wearing one dress of simple nature, and दूसरे हमारे सिर्फ सिंपल सादे कुर्ता पजामा, और किसी ने अपना कुर्ता पहन के उस पर जैकेट ओढ़ ली। The only thing is everybody is wearing different, representing different culture, representing different Veshbhusha, but everybody is towards the health. And if I say the theme of this today's title is Ethnopharmacology Beyond Times. I was just connecting what is beyond time. And if you just see द नॉलेज का जो पाई होता है, एक चक्र होता है, एक गोला होता है, उसमें 3 खाने में अगर हम डिवाइड करें, वन इज आई नो, एंड दिस आई नो इज नथिंग बट ईगो, कि हमको पता है, अरे यार तुमको कुछ नहीं पता, लेकिन वो भी हमारा 0.001% उस पूरे गोल का होता है जिस पे हम घमंड करते हैं, आई नो। हमारे समाज में, हिंदुस्तान में, ये आई नो काफ़ी बड़ा था, 1000, 5000, ये 1 लाख साल पहले, जैसे अभी हमारे आचार्य जी ने कहा। लेकिन हमने उसको अपने दूसरे देशों के इन्फ्लुएंस से हमने उसको भुला दिया। हमने कहा ये आई नो तो था, अब आई डोंट नो हो गया। उसके बाद एक दूसरा पाई आता है, आई डोंट नो, ये हमको नहीं पता, और इसकी हम खोज कर रहे हैं, इसकी विज्ञान हम कर रहे हैं, इसकी खोज कर रहे हैं कि क्या ये ड्रग एंटी डायबिटिक में काम करेगी, कैसे काम करेगी, क्या एंटी कैंसर में काम करेगी, कैसे काम करेगी, क्या ये हर्बल ड्रग जो 10,000 साल से इस्तेमाल कर रहे हैं, अब हम इसमें पुनर्विज्ञान लेके आ रहे हैं कि इसमें कैसे और कैसे काम करे, कैसे मॉलिक्यूलर मैकेनिज्म से कैसे करेगी। दैट इज आई डोंट नो एंड नाउ आई एम ट्राइंग टू नो। लेकिन उससे बाद जो 80% इस पाई का हिस्सा है, वो कौन सा है? वो है आई डोंट नो व्हाट आई डोंट नो। हमको पता ही नहीं कि हमको क्या नहीं पता, और वही काम यहां पे आचार्य जी और स्वामी जी कर रहे हैं। हमको वो पता करना है कि जो हमको पता ही नहीं है, जब हमको पता ही नहीं है क्या नहीं पता, यही हमको पता करने की कोशिश करनी है। पहले अगर हम ये पता कर लें कि हमको क्या नहीं पता, फिर हम उसको देखें कि क्या पता करें। तो ये ज्ञान के चक्षु खोलना जिसे कहते हैं, अगर हमें आ गया, हमें राइट टाइप ऑफ क्वेश्चनिंग करना आ गया, तो हमारी साइंस आ गई। हम हर चीज में, हम साइंस जो हम कहते हैं, हम पूछते हैं क्यों हुआ। एक अगर आपने हिंदी में गाना सुना हो, ये क्या हुआ, पता है ना, कब हुआ, कैसे हुआ, वो साइंटिफिक गाना है। ये कैसे, हाउ, वाई एंड व्हाट इज द मैकेनिज्म, क्या ये काम कर रहा है कि नहीं कर रहा, यही साइंस है। और हम इसीलिए जब हम कहते हैं कि जब हमारे विज्ञान हैं, 2-3 तरह के विज्ञान काम करते हैं। अगर हम कोई हर्ब या कोई मेडिसिन किसी में ट्राई कर रहे हैं, तो हम पहले देखते हैं कि डज इट वर्क, क्या ये काम कर रही है। तो हमें एपिडेमियोलॉजी लेके आनी होती है। हम ये नहीं कहते कि हमने इस महाराजा को ट्रीट कर दिया, इस किंग को ट्रीट कर दिया, 5 लोगों को ट्रीट किया, नहीं। विज्ञान चाहता है कि कितने सारे लोगों को आपने ट्रीट किया, कितने लोग उसमें से ट्रीट नहीं हुए, कितने आधे ट्रीट हुए, और उसके लिए क्या क्या आपने इन्वेस्टिगेशन किए जो कि इंटरनेशनल पद्धति से उसको मान्य हो। दैट इज व्हाट यू रिक्वायर साइंस टुडे, एंड आई एम वेरी प्राउड कि इसमें एक टाइम था जब हम आज से 20-25 साल पहले स्वामी जी से और बालकृष्ण जी से मिले, लोग चैलेंज करते थे कि ये डायबिटीज ड्रग ठीक करती है। स्वामी जी ने कहा कि ठीक करती है, लोगों ने कहा नहीं करती। फिर इसका जवाब एपिडेमियोलॉजिकल दिया गया, क्लिनिकल रिसर्च से दिया गया, और उसके बाद अब हमने मोस्ट मॉडर्न तरह के, चाहे इन विट्रो फार्माकोलॉजी हो, चाहे इन वीवो फार्माकोलॉजी हो, चाहे वो HPLC हो, चाहे HPTLC हो, मोस्ट साइंटिफिक, मोस्ट मॉडर्न इक्विपमेंट जो यहाँ पे पतंजलि रिसर्च फाउंडेशन में, जो मैं भी बहुत बार देख के सराहना कर चुका हूँ, वहाँ से हमने प्रूव किया। अब किसी को ये कहने की आवश्यकता नहीं होगी, हिम्मत तो लोग कर देते हैं, दुस्साहस भी कर देते हैं, उसको हम नहीं रोक सकते, लेकिन आवश्यकता नहीं होगी कि कहें कि साहब इसको दिखाओ करके। और इसका एविडेंस है, अगर आप जाके यहाँ के देखें लाइब्रेरी में, यहाँ की जो पब्लिकेशन का रिकॉर्ड है, मोर देन 700 इंटरनेशनल पब्लिकेशन फ्रॉम Patanjali Research Foundation. I salute those scientists working here. I salute those technicians working here. And I understand there are more than 250 patents here. There are more than 200 monographs, जो कि पिछली बार हम आए थे तो बालकृष्ण जी का एक जन्मदिन था, उस पर उन्होंने दिखाया 200 मोनोग्राफ। एक एक मोनोग्राफ लिखने में लोगों को ज़िंदगी निकल जाती है, उसका लिटरेचर कलेक्ट करने में। और ये सामूहिक collective effort, which I can say, have now you can go and get any reference of any of these of the products. That is what science is. And it is important when you look for the herbal drugs or ethnopharmacology, botanicals, everybody talks of standardization of the product. वो कहते हैं कि this product requires standardization, कि आज गर्मी में जो बना, सर्दी में जो बना, पत्ते जब छोटे थे तब तोड़ लिए, पत्ते जब बड़े थे तब तोड़े, और किसी ने कहा 7 पुट्ट लगाया, सहस्त्र पुट्ट लगाया, 100 पुट्ट लगाया, इनमें क्या फ़र्क है? Therefore the standardization is the global requirement. And I am very happy and proud to say that now today we have 500, more than 500 plants products का fingerprint है. Now if you have a 500 plants का fingerprinting, that means you can identify whether there is a purity 90% or 99% or 99.9% or it is 0%. And that makes a difference between the product which you sell from the Patanjali products and the other products. And that is the claim what you have. And that is, we are proud of that. Science is always evolving. And that's what we say, जो आज से हमारे पास 100 साल पहले हमारे पास प्रोडक्ट्स थे, चाहे वो च्यवनप्राश हो, चाहे वो कोई अष्टरिष्ट हो, कोई रिष्ट हो, चाहे कुछ भी हो, but then the formulation change, the improvement is the constant. Improvement change is the constant. If there is no improvement, there is no constant, and there is no constant, there is nothing dynamism. And that's why I'm proud to say that I was told by Anurag, 80 new formulations have been developed. 80 new formulations, whether it is for diabetes, whether it is for cancer, whether it is for pancreatitis, and whether it is for arthritis. अभी शायद वो फॉर्मुलेशन भी आ जाएगा जिसमें आप गंजे के भी बाल 1 महीने में उग जाएंगे. लेकिन अभी हमने ट्राई नहीं किया, इसलिए हम कोई कहते हैं. अभी हमारी मैडम ने कहा अभी थोड़े और कम हो जाएं, फिर बट लेकिन ये कहना आसान है कि हमने फॉर्मुलेशन बना लिया. द मोर इम्पॉर्टेंट थिंग दैट यू हैव टू हैव ए क्लिनिकल वैलिडेशन, you have to have a clinical trial. And I am very proud to say that now there are so many scientists here. There are scientists, more than 700, 300 scientists working here, 100 PhD scientists working here, and there are scientists, more than 50 scientists working, coming from industry, pharmaceutical industry. There are scientists who are collaborating with the different academic institution, AIMS, and collaborating for clinical trial, developing new protocol. These protocols are acceptable by their homeopathy, allopathy, or different disciplines, jahan kahan pe bhi ho. And as per regulatory requirement. The objective criteria, the end point which you set up for these clinical trials are those which are acceptable globally, those which can be published with international journals. And therefore, it becomes an evidence which is acceptable by science, by public, by regulator, and ultimately by the consumer. Ultimately, हमारा जो acceptability होती है, वो consumer की acceptability होती है. Any product, whether of any pathy, will not remain for longer period if this is not effective to the consumer for long. वो महीने, 2 महीने, साल भर करके बंद कर देगा. But if it is effective, it will go across the boundary, crossing the nation, and that's why you see the product which are Ayurvedic. I am not naming this institution's name, it is understood, is regarded, respected everywhere in the world. And that is what हमको उसको बढ़ाना है. हमें ये भी दिखाना है कि हम नई खोज करते हैं. And if you just go, I wish that those who have come from outside, they must go and see Patanjali Research Foundation, the clinical trial facility, the outpatient department, the systemic documentation, and the systemic analysis, statistical analysis. उनके जिसे कहते हैं, हिंदी में बोलते हैं, those who understand Hindi, चक्षु खुल जाएंगे. They will be mesmerized by the richness of the equipment. They will mesmerized by the richness of the rigor of the science which is being taken here. And that is what we look for, scientific evidence in this. And I am very happy that when ethnopharmacology, our Pulak Mukherjee and his society and international people have thought that let us go to Patanjali Research Foundation or Patanjali University. And it will not be surprising if they say after end of this meeting कि अब तो हम हर 2 साल के बाद पतंजलि में विजिट करना चाहेंगे, to see the progress, to see how it is working. Lastly, I would say, whenever sometimes you speak slightly longer, आपने देखा होगा ना भारत में लोकसभा में कई लोग कहते हैं कि मेरा माइक बंद कर दिया इसका मतलब है that means the time is over. But I am very happy that there are several organizations which are doing MOU with this university, and I am sure that when you ये लोकसभा के स्पीकर ने आदेश दिया कि भी आप आधा मिनट बोल सकते हैं, हमारे 2 स्पीकर उपाध्यक्ष बैठे हैं सामने, इनके आदेश इन्होंने फौरन दिया कि बोलने दो उनको आधा मिनट और, तो मैं बोल रहा हूँ. तो ये एक I think we all should feel pride that we have gathered here for international meeting, not only to discuss what is being done, what should be done, but also importantly to showcase to the whole globe that we are number 1 in Ayurveda, we are number 1 in the world in exploiting the natures for the benefit of the human being at the cost which is affordable. Affordable cost आप समझते हैं ना कैसे हो सकती है, जहाँ पे अमिताभ बच्चन और बड़े बड़े दिग्गज लोग करोड़ों रुपए लेते हैं एडवर्टाइजमेंट के लिए, यहाँ तो हमारे स्वामी जी खुद ही एडवर्टाइजमेंट कर देते हैं एक कपड़ा पहन के. और इतना इफेक्टिव एडवर्टाइजमेंट इसलिए नहीं होता है कि उनकी शक्ल ऐश्वर्या राय से अच्छी है या उनके कपड़े बहुत अच्छे हैं, इसलिए कि वो बोलते हैं उसमें साइंस होती है, उसमें जन साधारण को ठीक करने की अभिलाषा होती है, एंड दैट क्रिएट्स मास अपील, एंड दैट इज व्हाट मास अपील का रिस्पेक्ट रखना हमारे जैसे साइंटिस्ट का धर्म है. टू कीप दैट full stop, that to keep that respect on our system is the responsibility of scientists, clinicians, clinical pharmacologists like us. And therefore, we assure you, Swamiji and Balkrishnan ji, that we will leave no stone unturned to prove what is correct, to prove what is valid, to prove what is effective, and to prove that the product which is made in India will match the global standards. Thank you. Jai Hind, Jai Bharat.
Thank you very much, sir, for this excellent overview of the journey made by Patanjali Research Foundation and making us feel proud of the achievement made till date. I would like to thank you, sir, with a clap. Before proceeding further, we would like to humbly welcome our respected Pujya Sadhvi Dr. Devpriya ji to this symposium, Ethnopharmacology Beyond Times, and request everyone to loudly clap their hands welcoming her. With this, we would like to proceed to the next lecture, and I would like to invite Professor Mukherjee to kindly come on the dais for presenting the main objectives of this symposium. Professor Mukherjee, sir, is serving as a full professor in the Department of Pharmaceutical Technology at Jadavpur University, Kolkata. He is also a former director of the Institute of Bioresources and Sustainable Development under the Government of India. Professor Mukherjee has more than 29 years of experience in evidence-based ethnopharmacological research, and validation of metabolomics knowledge. He has published over 280 research papers and holds several patents. He is among the 2% listed global scientists published by the Stanford University and is a fellow of Royal Society of Chemistry, United Kingdom. I may now request Professor Mukherjee, sir, to kindly proceed with the objectives of this symposium. Please clap your hand, welcoming him.
परम पूज्य स्वामी रामदेव जी, परम आदरणीय बालाकृष्ण जी, and my friends from abroad, and respected Professor Y.K. Gupta and other colleagues from India who have joined to this Plant to Patient Series 4, being organized by Patanjali Research Foundation and the University of Patanjali as a satellite symposium for this 13th International Congress of the Society for Ethnopharmacology. Friends, it is my great pleasure to be associated with Patanjali Research Foundation, specifically to know more, or rather enrich my own knowledge with the Ashirwad, the blessings from Swamiji and Acharya ji. Thank you. So, जो प्रोफेसर Y.K. गुप्ता जी वो हमारा भी गुरुदेव है. So, he is my teacher. So, what he told, no, that is the main thing. So when we talk about plant to patients, India's journey from ancient root to modern civilization, that has emerged in a very big way. And that is what Acharya Balakrishna ji in his talk, in welcome address, highlighted what Patanjali has done. Tomorrow you will be hearing the history. But India's rich heritage of traditional medicines represents a unique intersection of culture, biodiversity, and healthcare wisdom. And this is to maintain an unbroken continuum of the nature-inspired practice, which is deeply enrooted to this empirical knowledge and for thousands of years. Yes, we talk about validation, evidence-based validation, and so many things. I was just talking with morning, no, we are having a lot of discussions with Dr. Rudy Bauer and my good friend Dr. Domenico. Yes, we need validation, and at the same time, we have thousands years of evidence already in hand. So you have to give value to both. Yes, we need to prove what thousands years we are doing, that is true or not true, but that doesn't mean that thousands year knowledge in any way wrong. If we cannot prove it, that is, we are really, we should be blamed for that. That हम लोग prove नहीं कर पाया वो thousands years का history जो है, हम लोग का culture जो है, healthcare knowledge जो है, वो उसमें scientific validity नहीं है. इस तरफ नहीं कुछ अभी तक तो कुछ नहीं मिला. And Patanjali Research Foundation, yes, here only we got this ancient wisdom translated with scientific validation, with technological integrations, and they are showing a global leadership for this evidence-based traditional medicine model. You have already heard from Dr. Y.K. Gupta that this research foundation, which was founded in 2017 in the spiritual capital of India, the Haridwar, with the esteemed leadership of respected Swamiji and Acharya ji, and inaugurated by our Honorable Prime Minister of our country, Sri Narendra Modi ji. The whole thought is, कैसे ancient wisdom का साथ modern science को bridge बना जाए. And really, the publications, the patent, the work, and this healthcare, wellness, product development, everything prove that the things are moving to the direct vision on that to be the global research foundations of the world in traditional medicine. And we feel proud with this core objective of this foundation, which is again the objective of our whole discussions on ethnopharmacology beyond times with this satellite symposium, is that evidence-based validations of this traditional Indian medicine, and specifically development of global standards for Ayurvedic formulations, conservations and sustainable utilizations of the medicinal plants, and creating waste to wealth models for the agricultural sustainability, and many more to prove that this is a global centers for evidence-based validation of Indian traditional medicine especially. Yes, this here is the essence, whole India, the Atmanirbhar Bharat, and the startup and so many things are coming up. I do not know how many startup has come up from Patanjali Research Foundation. It may be thousands or 100,000 more, because anybody, everybody जो इधर education लेता है वो they are having their own startup, they are having their own mindset. So we are very much grateful to Swamiji and Acharya ji for making India proud. We all feel proud that tradition का translation जो real sense में होता है ये पतंजलि में ही होता है. And in fact, without translation it is not an option now, it is an obligation. And when we talk about this ethnopharmacology beyond time, this is one of the major things that कैसे वो tradition को translate करा जाए and इनको scientifically validate करा जाए. So this goal is not to defend traditional medicines, but to strengthen it, and it requires the tradition survives by the wisdom and also by evidence, and we are translating the demand based on the regulatory aspect sometime. And here itself, the answer is with Patanjali Research Foundation. And that is why we from society and everybody involved, including Domenico, our International Society Vice President is here, Rudy and other friends, whoever is here, everybody, we thought that the Satellite Symposium, this itself will showcase that how the tradition to translation is happening. And Patanjali Research Foundation is a example, real example for that in India and throughout the world. Here, drug discovery development, safety, toxicology, pharmacokinetics, bioavailability, anti-infective research, anti-aging geriatric research, metabolic disorders, nanotechnology, environment health, in different area is being focused to establish that how the molecule work and how the molecules together work. So दोनों का reductionist approach, synergistic approach, everything together is being explored in this Patanjali Research Foundation, for which we really feel proud that how this translation is going on. Now, the whole thought of our this year Congress in Naipar and also in this satellite symposium, that is why we start from here, that all our friends or the scientists and the students who are studying here, that when we talk about the translational ethnopharmacology, a Patanjali Research Foundation ही है जिसमें real sense में translational ethnopharmacology का काम हो रहा है. And this traditional, this translational ethnopharmacology approach on 4 pillars. One is the traditional knowledge documentation, ethnobotanical, ethnomedicinal evidences drawn from living tradition and the historical text. I hope all my friends from abroad and India has visited the research center and also the herbarium. Dr. Bedpriya Acharya and our friends, Dr. Anurag, they have been taken a very great initiative. And so the first pillar, you will get all answer of that if you visit that place where the documented ethnobotanical and ethnomedicinal evidences drawn from living tradition and historical text. It is already evidence and from publication, patent and anything and everything what is there. Second aspect is the mechanistic validation. AV in vitro, in vivo characterizations or the bioactive constituents and molecular targets. It has been again proven in many, many different ways from our ancient Ayurvedic text by Dr. Anurag and his group in this Patanjali Research Foundation. Third pillar is the clinical relevance, what Dr. Y.K. Gupta ji was highlighting, that evidence of therapeutic efficacy, safety, pharmacokinetics in human population. Yes, there is a very good group in this research foundation, and also they have collaborations with many institutes, organizations in India and abroad, and you will be hearing some of those stories from Dr. Ray and from AIMS as well. And the fourth, the most important pillar is the healthcare integration. What Pujaniya Swamiji was highlighting, no, that integrative research, it happens in India first time in Patanjali, And we are so proud that Integrative Hospital has been opened last month. It was inaugurated and it is working in a very big way. So integration of this ancient and modern healthcare, including this regulatory acceptance, clinical guidance, incorporation, patient-centered application, everything is possible with this traditional ethnopharmacology. So friends, we talk about systemic ethnobotanical documentation, cross-cultural patterns or disease indication mapping, indigenous knowledge protocol, everything is possible through that, and which is again getting much more emphasized to the AI renaissance, modernizing the traditional medicine. This is another highlights what we are going to put on this digitizations of this ancient knowledge, predicting the hubs and the mechanisms with network pharmacology AI model, accelerating the natural product discovery, where the deep learning cheminformatics models will be provided, and also the quality and safety intelligence, including the sustainable bioresource management with remote sensing, AI forecast over harvesting risk and all these things towards precision and traditional medicines concept. So at the end, we all want to highlight that plant to patients, where was the tradition and what is the trend. So trend has been set up by Patanjali Research Foundations for the translational research and also what we think ethnopharmacology beyond time. And we are really very much grateful and thankful for the enormous contributions of the scientists, technologists involved in this Patanjali Research Foundation for bringing together this evidence-based Ayurveda with different techniques, where Society for Ethnopharmacology and the International Society for Ethnopharmacology is very happy to join. As you know, this society was rather established with the mentorship of our most respected former president of our country, Dr. APJ Abdul Kalam, who was the mentor, and with the mission to build a scientific bridge that honors traditional wisdom and uphold the rigor of modern validation, established in 2012 with this main motto of globalizing local knowledge and localizing global technology, vocal local. And that is our main aspects on that. And in fact, this society has a long presence in India with 18 local chapter, over 2,000 plus members, 12 of very impact studies we have performed, where 50 plus global knowledge outreach program and 15,000 delegates who join. We are having many programs, including the traditional healers program, which is one of the major thing, where we promote the Lok Sasta Parampara. We have the codified system, but the non-codified system, how to promote. And that is one of the major thing we took forward on that, to highlight on that. Yeah, and a very special thing what happened in this year with the respected blessings of our Swamiji and Acharyaji. We have been able to establish this journal with the system Ethnopharmacology and Sustainable Bioresource from Elsevier, and Dr. Anne-Marie Pardon, she is here, who has made all these things in a very different way to establish that it is a first of its kind international journal dedicated to the holistic, evidence-based and sustainable aligned research with the bridging ancient wisdom with modern science. It is a new home for integrative research. And this is the first journal ever established in India by Elsevier to a society. So we are very much grateful and thankful, and Anne-Marie's first visit is here in Haridwar. And we believe that this journal with the blessings of Swamiji and Acharyaji will be able to cope up with the advances in a new paradigm in ethnopharmacology that connects plants, formulations, people, culture, ecosystems, and policies within a single system framework, where we highlights everything, including the evidence-based validations, the bioeconomy development, the startups, the ethno-entrepreneurships, and many more. And also it aligns with the United Nations Sustainable Goals. So friends, we look forward that ethnopharmacology beyond times, and with this 30th International Congress, which we will be witnessing from 26 to 28 at Mohali. And also we have the Dr. Domenico will be highlighting, we are having In 2026, the IAC Congress in Italy, he will be telling more. And in 27, next year, we have a plan to host with over 50 countries together with different organizations of the world to highlight that how these translational traditional therapies can explore, educate, and empower, and how India can showcase the new route for development of these traditional medicines with these validations, which strengthen global collaboration, influence policy frameworks, advance evidence-based traditional medicine. So with this, I once again thank you very much. And in fact, for all our friends from abroad taking so much of pain, I know many of them, they just reached Delhi yesterday 2 p.m., 2 a.m. in the night, means today early morning, and they are here today. So we are very much grateful and thankful to all of you who have joined, and I'm sure that this bioresource, which are the nature's gift for everyone, through that ethnopharmacology and the evidence-based validations, we will be able to promote and those gifts for everyone for the healthcare, and also preserve those gifts for the betterment of the healthcare of the society at large. Namaskar, Jai Hind.
I would like everybody to clap your hands, thanking Professor Mukherjee for presenting the symposium objectives clearly and highlighting the tasks done by the Society of Ethnopharmacology.
It gives me great pleasure and honor to invite Dr. Anurag Varshney sir, Vice President, Patanjali Research Foundation, to the dais for presenting an introduction of Patanjali Research Foundation. Dr. Varshney sir has obtained his doctoral from NCPSTFR Bangalore and his postdoctoral experience from Yale University, USA. He has more than 15 years leadership experience in reputed pharmaceutical company like Renbixi and Daiichi Sankyo. I now respectfully request Dr. Varshney sir to share the activities and achievement of the Patanjali Research Foundation. Kindly welcome him by clapping up with your hands.
I would request all of you go in the same sync as the Om that I will try. I am not the best at it, but I will request if we can have a deep breath and have a chant out of the Om so that we can connect with the same cosmos that we are sitting here at Patanjali. For the folks who has come from abroad, it's an humble request. Try it out. It's very, very relaxing, and it really connects you with the theme that we're going to go through. So let's start it. Thank you very much. So with my humble regards and bow in the holy feet of Param Pujya Swamiji, Param Shraddha Acharya Ji, I would like to welcome all of you to this fantastic 1.5 day symposium on ethnopharmacology beyond times. And I'm very happy to have the lot of guests come from abroad who have taken their time to come and visit Patanjali, and I would like to welcome them by extending the heart feelings from Patanjali Research Foundation. And we have with us Dr. Domenico Delfino, who's sitting in the audience and who has been here earlier as well, a couple of years back, and we have been connected for quite a while, and we have been conducting some good research together as well. So let's clap for Dr. Domenico, who has been a fantastic colleague, fantastic scientist, and has been the present president of the International Society of Ethnopharmacology. And we also have Professor Louis Felix Notias, who's sitting in the audience right over here. He looks very young, but full of energy, and has been exploring Patanjali for last 1.5 day, and he has been liking it very much too. So welcome, sir, to have you here with your mother, who's a fantastic photographer as well. And then we also have Professor Theodrat, who has come this morning at 2 p.m. from Thailand, and I think she's somewhere in the audience over there too. So thank you very much indeed for being here and being a part of this meeting. And then we have a couple of big stalwart of ethnopharmacology across the world, Professor Rudolf Bohr, sitting in the audience over there with her better half. Let's clap for him too, to welcome here at Patanjali Research Foundation. And then Professor Annie Mary Porden, who comes with a fantastic experience of working with a large publication house, Elsevier. Let's clap for him. And then we are missing two of our colleagues who could not come because of the visa issue they actually had it. We had Professor Jang Honji coming from Hong Kong. His visa got stuck at the nth hour. And we had Professor Satish Chandrabachar from Bangladesh, and he could not actually travel because of the political issues between those international borders. And then we have our mentors, Professor Y.K. Gupta sir, and I'm very thankful to him that he started the whole meeting so beautifully. Whatever introduction I wanted to give about Patanjali to our overseas guest, I think he covered most of it. So I would really like to thank him and would like to clap it as hard as possible. I think the students on the back can take out their hands from the phone and clap it faster, right? So that is for sir. And then we have Professor Pulak Mukherjee, who just set up the tone that what we're going to see. And then we have the audience that's sitting here, all the professors, and we have Sadhvi Depriyaji with us, Professor Sanjay Chachakji, who's going to host a much bigger meeting, hopefully sometime soon. And then we're going to go through Dr. Depriyaji, my colleague from PRF, and then professors from university, professors from Ayurvedic College, and all the students from BAMS and BNYS course. All of you are here, and I'm very thankful. On behalf of the institution, I would like to extend the warm welcome and take it forward. So let's clap it for yourself, and then I'm going to start giving a little bit of introduction. So what I'm going to do today, I'm standing on a very tall platform, thanks to Acharji, because he gave that bridge to us so that I can connect. And I also see our respected Pujya Swami R. Devji in the audience, and I'm hoping to see Swami Bajrang Ji as well, both the service heads of Patanjali Wellness and Yogram, who has been doing fantastic things. So Swamiji, welcome for you too in this meeting. So what I'm going to give today, the audience who has come from outside and who are here for the first time in Patanjali, and those of you who have been part of it, for you it may be a slightly revision kind of thing. Take it like that when you take a flight, जब आप हवाई जहाज में जा रहे होते हैं तो you get a lot of security and announcement by the air hostess. You put a seat belt, do this, that. So for some of you, this might be like that security announcement. It is repetitive, but it's important. So please go by it, and I'm going to explain to you that what we do, how we do, what's the plan we have, what we have achieved, and which way we are going to go. I do speak sometime faster, sometime I also stammer, so I request all of you to bear with me, and if I get too fast, raise your hand, I'm going to slow down a bit. And we do have Professor M.D. Ray in the audience. I welcome him as well. So we have also planned three things here. So we are conducting clinical trials at three big locations. One is the AIIMS Rishikesh, there's one in AIIMS Delhi, which is just completed, and one in Tata Memorial Center. Other than that, we are also running 16 clinical trials. So these trials are going on. So we have the PI for those trials here, Professor M.D. Ray, and his better half is here. We would have Dr. Puneet Dhamija tomorrow, and we will also have Professor Bikram Gota from Tata Memorial tomorrow, who will then talk about what is their experience has been and how the drugs, the formulation which have been developed by Patanjali, being doing good in the clinical trial run by them. So let's welcome all of those as well whenever they are here. So to introduce PRF, Patanjali Research Foundation, we are blessed, influenced, guided, and directed by Param Shraddhe Swamiji, Param Pujya Acharji on the daily basis, and on that direction the PRF have been started. This whole setup started in 2017 and inaugurated by our respected Prime Minister Modi ji, and these labs have been set up to bridge the gap between the modern practice and the ancient medicine knowledge Ayurveda. They're probably one of the only kind, literally state-of-the-art facility to conduct the research on Ayurveda and yoga. Top-of-the-line equipments, globally trained workforce energize these operations on a daily basis. So we have like close to 300 scientists, and most of the scientists, the biggest toys that we want to have, जो हमारे बड़े खिलौने हैं, Those toys are the equipments. If we have a good technology and good equipment, we can do wonderful science. So we are very blessed, and we really feel very fortunate from that we get a lot of those toys to play with, thanks to the guidance and support from Param Puj Acharya and Swamiji, that we can do those experiments, and all of those equipment, all of those thoughts take the science in the right direction. Now, like any other institution, Patanjali Research Foundation has a mission, and the mission is to improve health and quality of life through research and innovation. That have been the mission of the institute, and we have been working very strongly about it. And now to do this, there is a vision that we really follow. So we already have an unstructured Ayurvedic and herbal information combined with the rich history of medicinal plant. And if you put a funnel of those scientific methods and combining the modern practice, larger scope of delivery, as well as the multi-pronged way of dealing with it, and go something about what we would like to do, a scientifically validated, evidence-based medicine system, what is called Ayurveda, which is accepted beyond the national boundaries of India or Southeast Asia, where Ayurveda is accepted, respected, and practiced as a mainstream medicinal system. That is the goal we are heading for. That's the vision that we're going to take it for. So I will request all of you for that vision, for that mission. Let's clap it hard. I'm going to clap with you. Now, when we talk about the research, and this is our main meeting that we're going to talk about, the research strand that Patanjali has, the strand that are there, which are there at Patanjali Research Foundation, the accreditation that we work with. So whatever is required to conduct a research in India and beyond, whatever the qualification, the quantifications, the regulatory compliance are needed, those are existing at Patanjali. This is a slightly busy slide, but I would like to take your attention towards that our laboratories are anyway accredited. They are accredited with DSIR, which is a government institution, Department of Scientific and Industrial Research. More than 300 scientists work here, several PhDs and postdocs with global experiences. We have a full-fledged animal house as per the CPCA requirement, governed by Institutional Animal Ethics Committee. We have herbarium at PRF, which is accredited by New York Botanical Garden, USA. Our laboratories are in the compliance with Biosafety Level 2, which are again governed by the Institutional Biosafety Committee, and which have a dedicated clean and service corridor for microbiology and experimental animal facility too. On the top of it, we do a lot of clinical trials. We have two large clinical facilities in Patanjali Wellness and Patanjali Yogram. On the top, we do multiple trials at external sites. Some of them are happening in India. 3 of them are happening outside India as well. Most of them are driven by Institutional Ethics Committee for Human Clinical Trials. And all the trials we conduct have been registered with the Clinical Trial Registry of India, what is called CTRI. And then what the good freedom we have, what the good liberty we have, our next door we have 250-bed Ayurvedic hospital with NABH accreditations, and which is an approved Ayurvedic medical college. Students are sitting here. You guys can clap for yourself as well for all the folks coming from this fantastic Ayurvedic medical college with the PG approval in the 8 subjects, and 2,200 Ayurvedic doctors across India at all those Patanjali Chikitsa Kendra, which has a daily footfall of close to 15,000 patients in a day. Now, see these numbers, 15,000 patients in a day, and if you combine them together, it comes to close to a 1 crore, 35 lakhs. Like, we are talking about 13 million patients' data are there in Patanjali Electronic Medical Record, which is a humongous information. And as we say, data is money, data is information, information is future. So when you talk about this much of information about the patients and their treatment and all kind of modalities which have been all combined here. So this, if you see this whole slide in total, you would see what we have, a diversified research framework. That means that whatever is required to conduct the research, ethnopharmacology, herbal chemistry, herbal biology, whatever name we would like to take it, it actually either met here or Patanjali Research Foundation or have been built by the technical competency that we have. So when we talk about this foundation, so we have different segments to it. So we have our herbal research division, which is led by my colleague, Dr. Betpiriareji. Tomorrow she is going to talk more about it and to explain the type of work that we do. That's, again, a fantabulous things. We have got the largest collection of plant paintings. We have got the largest collection of the compilation of medicinal information from the plants, which comes in form of a World Herbal Encyclopedia. We're going to hear about the tomorrow. We have plant paintings, we have a yoga research, and we also work on history and ancient documents. As Puj Acharji mentioned in the morning, that we worked a lot on history, how medicinal system evolved, how the things have changed from last several thousand years before. So those things are also very, very interesting and research driven. And we have our fantastic colleague, Dr. Rashmi Mittalji. She's going to talk tomorrow along with Acharji, and we're going to see the ancient documents and the history, what really happens. And then we get good support from New Product Development, Patanjali Ayurved Limited. We have Dr. Ram Gupta ji sitting here from the same. So thank you, Ram, for being here. And then from Central Labs, and also linked from Patanjali Hospital and Medical College. And this is what I'm going to talk today: drug discovery and development. So we have this as a very significant component of Patanjali Research Foundation, and that's where most of the folks who do a wet lab research at PRF work there. And I'm going to then introduce to you what we do here, what is the aim, and what are the modality we really work with. So these are just a glimpse of our infrastructure, the toys that I was talking about. So the PRF has, the drug discovery development has this 5 different components: in vivo biology, in vitro biology, herbal chemistry, microbiology, and clinical research. And the few of those pics that you see are right on the screen. And what we do, so when we started this whole operation, we had a long discussion with Param Puj Acharji, Param Shraddhe Swamiji, that how are we going to do. In Ayurveda, in the classical text, there are close to 1,000 formulations which are defined in the ancient old text. And there are close to 1,000 plants that we really work with. So if we combine them together, that becomes close to 2,000 test articles. Basically, in terms of research, you define everything you want to test as a test article. So there is a 2 way of doing it, that you take one test article, one drug, and start to look for the what disease it might work. That becomes basically a drug-driven approach or a formulation-driven approach. The other would have been to find a disease and see which formulation can work over there, what we call disease-driven approach, and we started working on the same line. Param Puj Acharji advised us, guided us, that let's go in this direction and find out if you go by a bucket of different therapeutic areas. For example, you look at inflammation, metabolic diseases, cancer, neuroscience, and infectious disease, and then you look for the type of disease you can work with and see whatever models you can set up. And then you test what are the formulation that you have with you, whether it's a drug working for diabetes, working for neurons, or any kind of pathology that you want to associate with. And these are a number, what you see are the validation that we have already done. These are the really, really large number. And if you look at the success rate of these experiments, they actually meet any of the laboratories anywhere in the world. So for this success, let's clap for the scientists who worked very hard on those. So we work on inflammation. These are the disease, diabetes, and other stuff come in the metabolic disorder, cancer biology, neuroscience, and infectious disease, where we work with the bacterial, fungal, as well as viral diseases. During the COVID time, we were the first one to show that herbal medicines has an capability to actually fight with the bigger monster, COVID-19, and the whole team has actually taken off from there. And in the leadership of Dr. Sabta Lochabji right now, we are doing a lot of work in this area. And then we have different kind of biology function too. Now, this is my favorite slide, and this slide personally drives me a lot, and all of my able team that works it actually get driven by that. So if you see, we know what is pharmacology. So when we talk about modern drug discovery, जो नए दवाइयों बनाने का तरीका है, जो मॉडर्न मेडिसिन में सिंथेटिक ड्रग्स में बनाए जाते हैं, जब फार्मा तरीके से बनता है, इफ़ यू सी दैट टू गेट अ ड्रग टू द मार्केट फ्रॉम अ सिंथेटिक रूट, you have to go this way. This is called conventional path, that you make a compound, you make a molecule which is made by medicinal chemist, you test in the animals or in any models, and then you test in the man, what we call a clinical trial. This path is a conventional path, which takes close to $10 billion, 10 billion, not million, $10 billion, and 10 years. And still there comes with lot of unsuccessful results and come with lot of adverse effect, the side effect, what we call it. And the chances of success are also very low. So how do you going to deal with the disease burden that our society has? The one way to it, to go reverse path. Go it in the reverse path. We then can get inspired by the traditional knowledge. which actually the routine laboratory to clinic reverts to clinic to the laboratory. Why is that? Because we know from our ancient science, from our ethno practices, that Ayurvedic drugs, herbal drugs work. We know when we are going through a pains, we take different medications. When we're having difficulty, infections and fever, we take tulsi, we take giloy, all of that work, and which have been there in the practice for thousands of the years. So we know that efficacy exists, which is the biggest lacunae, biggest bottleneck when we go for a conventional path. So that would mean if you know efficacy, then it's just a matter of validation. All you have to do, validate it, find out where is that coming from, and then set up the safety. When people talk about therapeutic index, that means the ratio between the dose you get the adverse effect and the dose where you get a pharmacological effect. That thing is in the favor of the synthetic, that thing in the favor of natural medicine, Ayurvedic drugs. So all we have to do is start from there, which is the most important starting point, and validate the efficacy. Then you go to a reverse path. Then all you have to do, we have a data from the man, you go to the mice or any animal model and go to the molecule and find what are the players which are controlling the biology, whether it's happening at the level of the compound or it's happening at the level of the receptors inside the cells, whether it's happening inside the cell, on the cell surface, how the signal is getting affected, intracellular versus extracellular, all getting linked. And that's where we are going to work with. And that's take to my next favorite slide, where we talk about how do we strike the balance. How do we strike this balance between much needed need of the society of drug discovery. So this is my favorite slide. As I said, this is the triangle where you can combine Ayurveda with modern science. That means all the scientific advances that has happened for last 200 years. If you talk about X-rays, you talk about ultrasound, you talk about MRIs, these are scientific advances. Allopathic system happen to use them slightly better than Ayurveda have used. But if you combine them together, along with the modern medicine, which actually focus on evidence-based clinical practice, and then you would be able to set up this golden triangle. Now, those of you who are still awake and listening to me, and if you focus on this slide, if you eyeball this triangle, if you see this, you will find this triangle looks impossible at this moment, because all of these three arms of the triangles are not matching. Those who have seen it, let's clap for them, because I have seen it, right? So if you see this triangle looks impossible, but that is achievable. And if you see deliberately, I have put the logo of Patanjali Research Foundation, my institution, at the heart of this triangle, because we strongly believe in the guidance of Param Pujya Swamiji, Param Shraddha Acharya, that we are going to meet this, reach it, and achieve it. Of course, Swamiji. So, okay, Swamiji. So traditional wisdom, research, and evidence-based medicine, and that's going to lead it to there, Swamiji. तो हम यहाँ से यहाँ पहुँचेंगे. तो ये जो पूरा पाथ है हमारा, वो यहाँ से ही पहुँचेगा. सो आयुर्वेदा में ये है, एविडेंस स्वामी यहाँ से करेंगे कनेक्ट और इस ट्रायंगल को पूरा करेंगे. सो आइडिया ये है कि जो गोल्डन ट्रायंगल अप्रोच है ड्रग डिस्कवरी की, वो आयुर्वेदिक साइंस से पूरी की जा सकती है. अब इस काम को करने के लिए what we do here at Patanjali Research Foundation, the whole landscape of the work that we do, we go from plants to product and from product to the patients, which is the theme of this meeting. That how do we start from the plant, how do we go towards the product, and what are the steps that we take, what are the ways that we do. And in next 3 to 4 slides, I'm going to go very quickly through this, the technologies we use, the paradigm that we follow, and the protocols that we have. What is the SOPs for doing research going from plant to the product? So we start with the herbal chemistry, where we find out which plant to work with, plant selection, authentication, plant extractions, and biocompound isolation. Then we go to analytical chemistry and try to find out what are the signature fingerprints and find out what are the things that can be done. This is being led by Dr. Pradeep Nanji, who is the head of this function, and he has been in the audience and trying to set up this conference as well. Then we go towards biology. We do in vitro biology work, where you work on the 2D cells, 3D cultures, do a lot of cell-based assay, mechanism of actions, and molecular targets being seen, which becomes very critical to find out how drug works. That is being done, led by Dr. Rishabh Devji, and supported by Milsi ma'am and also Sandeep ji, which are there too. And then we go to in vivo biology, which is a fancy stuff, which is done by Dr. Sandeep Sinha ji and his team, then where we work on the efficacy, predominantly to find out if a drug works in what model, how that happens. That is being done. And then we move towards formulation. If things are good, then we complete the whole cycle, generate a formulation and get it ready. And that's where the team from Dr. Nain comes back here, and we find out what are the requirements, what are the regulatory landscaping, and what can be done. In parallel, we also start the clinical research to find the mode of actions for the drugs, also the clinical relevance, which is being led by the clinical research team, and Dr. Momita ji lead that function. So I'm quickly go through each of those components. This is what we do for herbal chemistry: select the plants, find the solvent, get it done. Sometimes we do a supercritical fluid extraction where we use liquidified CO2 to extract the best part of it, take the oily formulation, find out what is there, and then go for marker identification. And then we also go towards analytical chemistry, where we do HPTLC, high performance thin layer chromatography. All of these toys and tools are there in our laboratories. Some of you who are interested could visit and see those too. From HPTLC to UHPLC, LCMS-MS QTOF to UHPLC QTOF, MSMS FID, which is gas chromatography, to MSMS, to find out what is there in the air, which is a form of the volatile component, and what's in the liquid, which can be seen here too. And from there, then we go towards in vitro biology. That's where slightly more exciting things start to happen. You start to see the interactions of the herbal compound with the biological things. So you have cells that you do 2D cell cultures. You do a 3D cell culture when you make spheroids on them, which are slightly closer to the intact organs. And then we also do ex vivo models. That means the cells isolated from the patients, which are also grown. And then we do a very detailed biochemical molecular characterization, gene expression, changes at the level of the protein, changes at the level of protein phosphorylation, and there are other kind of changes that happen in the cells. And then we find out what's the mechanism of action. That means if we have in formulation and extract, what it works and how that works. I learned a very nice word from Puj Swamiji, Karyakari Siddhant, which is mode of action. So Karyakari Siddhant, we start to see from here that how these things work and in what principle things start to move in the right direction. And then get slightly more dynamic slides here, where you get to see the different model organisms that we work with. So we work with the C. elegans, which is a very elegant worms, which is a distant cousin of earthworms, and we have a dedicated laboratory where we have close to 116 types of C. elegans with us, which has genetical modifications, and you can use them in a very good model system for the disease. We do that. Recently, we have set up a Drosophila lab as well, which is called Drosophila melanogaster. Easy language called fruit fly is a fly that comes to your banana when you leave it on the kitchen, and that is a very good system too. And zebra fish, which are small, tiny fish. We are now making a new facility where fish and fly will stay together, and that facility would be also ready very soon. And in animal house, we have mice, rats, hamsters, guinea pigs, and rabbits, and we work with them to actually set up different disease model. These are all being done at Patanjali Research Foundation in Haridwar in these labs. We also work on dogs that we do a third party in Hyderabad, and we are also started to work with the monkey models that we do in China. So basically, whatever the requirement for a drug discovery is being done, met or bait here at PRF. And then we go towards the formulation. Once we find something, then we do a product optimization, find out what's the best way to deliver it. Is it a capsule, or is it a tablet, or is it a spray, or it's a topical ointment? Depending on the disease need and the patient compliance, we set up those systems, go towards the regulatory compliance and find out what are the needed to have it towards the market, what are the documentation that must be done, test the stability, scale it up, and transfer to our fancy team sitting at Padartha for the commercialization. And then comes to the clinical research. So that becomes one more critical component where we test the efficacy and safety, monitoring the patients, and also measure the outcome assessment. Not only for the disease, we also measure for the quality of life. We also measure the disease-related health quality of life and see what kind of changes happening in the life of the patients when they're going through a disease, and when they have been given these medications, Ayurvedic formulation, how those changes are being done, that get messaged too. And one thing I'm very proud to say, and which happened very recently, the Patanjali Research Foundation has been now accredited as a certified clinical research organization, a CRO, which is the first in the Ayurvedic industry. This is the first in the herbal space. And we are very proud that the whole team that has worked for it to set up that PRF is now a CRO, and a lot of things can be done under this umbrella and this accreditation. When we do all of these work, what are the outcomes we get? So the two-way possible. First, we develop a product which goes towards a market, which help the patients, which makes sure that our society remain disease-free. The burden of the disease decreases. And what you see over here are those formulations. Close to 80 new formulations, 80 new medicines has been developed on the basis of research generated at PRF. So from head to toe, different organs, 5 vital organs, and 6 accessory organs if you see, from heart, brain, kidney, liver, lungs, everywhere, whatever the disease etiologies, pathologies possible can be addressed, treated, and cured by these formulations. Some of them have been really fantastic. Some of them has been totally novel. Some of them has been for the first time in the history of Ayurveda as well. For example, we have the formulation for pancreas, we have a formulation for prostate, which have never been thought of. And then Param Puj Acharya, Param Shraddha Swamiji has thought in those lines, developed those formulations, guided us in the right way of mixing those components and telling us that this would be the most optimal way of getting it. And I'm very happy to see that 95 times out of 100 times, that's a 95% success rate, most of these formulations worked. So Swamiji, Acharji, thank you very much indeed for giving us that guidance, giving us that enlightenment to finding out different formulations. So this is what you see, which is from different body organs. And if you talk about the different physiological state of disease, whether it's actually pain, from infertility towards immunity to fevers and other things, and not to forget the cancer, but which I'm hoping that Dr. M.D. Ray will talk more about it, that all of these formulation works in those areas too. So the team has worked hard to reach to that level that we can validate the every stage of the research, can find out what drugs work at what level, at what disease pathology etiology can be controlled and treated by these formulation too. So these are the one of the outcome we get from the research. The second outcome come from the high-end research paper. The drug discovery team alone has published in last 6 years 130 research papers with an impact factor. The total combined impact factor are close to 600. The average impact factor 4.5, which is really humongous. And I feel very proud standing here saying that this could not have been done anywhere else. As Swamiji says in the morning, that people who work on a single product, on a single drug or a single paper, they consider themselves that one PhD is done. Here, we are talking about 130 PhDs, or that too of the world-class work. So thank you very much, team, for being there and doing that too. And that comes to my final slide and come back to where I started from, the mission that we actually follow, that the mission that we have, we want to offer the whole world, the science of medicine from the oldest medicinal knowledge base, Ayurveda, and the mantra that we follow every day, Sarve Bhavantu Sukhna, Sarve Santu Niramaya. Thank you very much, team, and I really welcome all of you again, and I hope in next 1.5 days you are going to enjoy and learn. Students in the back are going to learn a lot from international speakers with the experts who are going to share most of the things which they have learned, and I'm sure when they go back to their home, they will remember us, and we will also remember them and take this thing together. And I hope it's a good starting for a much bigger collaboration at a global level where PRF would be a big part of it. So thank you very much indeed. Swamiji Acharya, aapke charanon mein rakhna.
Thank you, sir, for presenting the scientific vision, objective, and achievement of Patanjali Research Foundation. Now I request everyone to kindly please put your hands together for him.
Moving towards the keynote lectures, may I now request Professor Domenico Vittorio Delfino to kindly come to the dais for presenting his keynote lecture titled Uncovering Context-Dependent Immunomodulation, How Ficus rubiginosa leaf constituents influence the Treg Th17 axis. Professor Delfino is a distinguished physician and pharmacologist with over 30 years of experience in teaching and research. He is serving as the professor at the University of Perugia, Italy, and holds several other key important leadership positions. Currently, he is serving as the president and member of the board of directors for the International Society of Ethnopharmacology. Professor Delfino has published more than 106 peer-reviewed research papers and serves as associate editor for several international journals. I would request Professor Delfino to kindly proceed with his lecture. Thank you, sir.
Thank you. Thank you very much for this kind of presentation. And just a moment that we, okay, yeah, presentation. Presentation. Okay. Okay, ladies and gentlemen. First of all, I would like to thank very much the organizers for inviting me. I am indebted with Anurag for this, with Swami G and Acharya G. Thank you very much. You know, this is my second time in Patanjali, and I am so happy to be here again. I'm really happy because in the audience there are a lot of young people, a lot of students. I would like to be, yeah, I would like to be there with them, but I guess I'm too old. So anyway, with Anurag, Patanjali, and my university, University of Perugia in Italy, we have an MOU now, and so we hope that some students from Patanjali University can go to visit Perugia, our university, and maybe, Anurag, maybe this year someone can be, our student can be here. Okay. So what I'm going to do today is to tell you a story about a family plant. The family is Morace family, and to tell to you all this story, I would like to bring you with me for two trips, two travels. One is from India. Just a moment, please. The presentation.
Yes, sir, it's work.
It's better if you put in the desktop.
Okay. Yeah, it's a desktop. It's on desktops.
Okay, it works now. So what I'm going to do is to bring all of you with me for two types of trips. One is from India to the Far East, to the Vietnam, and the other is from India to Italy, of course, where I am from. This These are the two trips. So let's start from going to Vietnam to talk about this plant family, the Moraceae plant family. One species in the Moraceae family is this tree. The name is Artocarpus tonkinensis. Is a Vietnamese tree, is endemic of the north part of Vietnam, where they know this tree as an ornamental plant. For example, a plant that is present in the garden of Buddhist pagodas. But in the very north side of Vietnam, close to the border with China, in a mountainous area, this plant is known as a medicinal plant because they use the decoction of the dried leaves of this plant as a treatment for arthritis and backache. This was discovered by Dr. Pham Truc Lam during the war of Vietnamese against France. And the people that use the decoction of this plant for treatment is an ethnic minority, around 800,000 people living in that mountainous area around Son La, Lai Chau, Sapa, Cao Bang, Lang Song. And we know 5 branches of this ethnic minority, the Hmong, and they are called based on the colors of their dress. For example, white, red, green, flower you see in the middle, these very colorful dresses. They are dressed by the flower monks. And this decoction of Artocarpus tonkinensis is used by the black monk to treat arthritis and backache. So we made some experiment to see if we can validate this knowledge, as Anurag said before, in a reverse pharmacology, let's say, pathway. And we have a very nice model, mouse model for arthritis, which is the collagen-induced arthritis. Here you see my mice. This is an Italian mouse we call mouse Gigio, and we love it very much. So I gave to him this treatment because I was certain that was a safe treatment. Anyway, when you treat the mice with collagen and then you challenge again with collagen after 28 years, these mice develop arthritis. And they develop arthritis because they produce Th17 cells that goes by the attraction due to the chemokine CCL20 to the joints, and also because macrophages become osteoclasts that destroy the joints, bringing to arthritis. So Th17 cells and osteoclasts are very much central for the pathogenesis of arthritis. So we gave to these mice the decoction of Artocarpus tonkinensis, and as you can see here, their swollen joints, paw, were completely reversed by the treatment with the decoction. And also at the histology level, if you can see, in a healthy mouse, you have a very smooth cartilage and no inflammatory infiltration in the joints. But if the mouse develop arthritis, the cartilage is broken and you have a lot of inflammatory arthritis. If these mice drink the Artocarpus tonkinensis decoction, you can see that again the cartilage is smooth and you have only a small inflammatory infiltration. So this decoction is really useful, as demonstrated by these experiments. We also made a gene expression profile of the joints, and as you can see here, you have a complete shutdown of interleukin 17 and interleukin 22, a downregulation of interleukin 6, of CCL20, the chemokine, of CCR4, the receptor for that chemokine, and interleukin 23. All these molecules are characteristic of a type of cell, which is the Th17 cell, which we have seen before is important in the pathogenesis of arthritis. That the decoction was going to inhibit the development of Th17 cells, we could see also by analyzing the pose, the lymph nodes and the spleen of that mice, as you can see here by real time. There is a significant downregulation of interleukin 17 and the interleukin 22, the two cytokines characteristic of Th17 cells. So we can conclude that the decoction of Artocarpus tonkinensis leaves improves signs, symptoms, and histology of arthritis, since it inhibits the development, the function, and the migration of autoaggressive Th17 cells. As you can see here, the Th17 cells are inhibited in their development, in their function, and also in their migration from spleen to the joints. We also studied the osteoclasts, and as you can see here, also the osteoclasts were inhibited in their development. We studied then an in vitro model of osteoclasts, and as you can see, the decoction was going to inhibit the development of TRAP-positive cells, which are osteoclasts, and also the development of markers of osteoclasts, such as ACP5, CALS-R, MMP9, and CTSK. And also another feature of osteoclasts function, which is the phosphorylation of SARC. Also, this was the action of the decoction in vitro on osteoclasts, but also in vivo, all the markers of osteoclast generation were shut down, as you can see here, ACP5, MMP9, CTSK, and calreticulin. Okay, we made also a fingerprint, and as you can see here, we have many compounds in this decoction. Mostly these are auronal glucosides, and the most abundant is mesopsin 4-beta glucoside, T82, which also have the same features of the entire decoction, but it was less stronger. This means that it is able to inhibit the development of Th17 cells, but in a minor way compared to the entire decoction. So the decoction is better of the single main component of the decoction. This is true for the development of Th17 cells, but also, as you can see here, for the development of, for the inhibition of osteoclast generation. Th82 is effective, but not so much as the entire decoction. Okay, so we can say that collagen in our mouse model can induce the development of arthritis because it induced the development and function of Th17 cells from spleen and the osteoclast from macrophages. And this brings to arthritis. But if you give the decoction of Artocarpus tonkinensis, you completely inhibit the development of Th17 cells and osteoclasts, and the arthritis reverses back, and the joints are again healthy. Sorry for this. So the take-home message for this first part is that Artocarpus tonkinensis decoction has a mechanism similar to that of very innovative biologic drugs, such as, for example, monoclonal antibodies against interleukin-17. And this decoction may be useful for other diseases that have the similar mechanism, such as arthritis, for example, multiple sclerosis, psoriasis, and the asthma resistant to glucocorticoids. Now let's go back to Delhi, and from Delhi let's go to Italy, to Rome first, but then we go to the South Italy, to the Sicily. Sicily is an island, very beautiful, and the main city in Sicily is Palermo. Palermo is an ancient and a modern city at the same time. Very beautiful, one of the most beautiful cities in Italy, as you can see here from many monuments. And also is an ancient city because they have ancient religious traditions, such as also, you know, similar to India in some way. Here you can see people that for a religious ceremony, they undergo a complete trance. It's something that can happen only in that place. In this case, for the celebration of Easter. But Palermo has also a very prosperous vegetation heritage, and a lot of morace trees are present there. For example, a ficus here on the left, you can see a symbol of Palermo, which is an ancient ficus, and in the middle you can see another type of ficus, another species of ficus, is Ficus rubiginosa, that you can see here. Also, Ficus rubiginosa belongs to the Moraceae family, and we, you know, thought that maybe, as for Artocarpus tonkinensis in Vietnam, another member of the Moraceae family can, in some way, influence the immune system of our organism. So we made an extract from the leaves of Ficus rubiginosa and tested them against to see if they influence in some way the life of T lymphocyte, of CD4 T lymphocytes. Now, CD4 T lymphocytes derive from thymocytes, they go in the spleen or in lymph nodes and under a basic type of lymphocyte, which is Th0, and there they differentiate to different subtypes, Th1, Th2, Th17, T regulatory cells. We can do this also in the lab by stimulating the activation of Th0 cells through antibodies against CD3 and against CD28. And then we can drive the differentiation of Th0 towards the different subtypes, for example, Th1, Th2, Th17, and T regulatory cells. This is done by different cytokine cocktails, for example, interleukin 12 plus interleukin 4 to get Th1, interleukin 23 plus P40 to get Th2, and TGF-beta and plus interleukin 6 to get a balance between Th17 and T regulatory cells. So when you, in this experimental setting, you block the differentiation towards Th1 and Th2, you get only a balance between Th17 and T regulatory cells. Th17 is characterized by a transcription factor which is called ROR-C, and Treg cells are characterized by another transcription factor, FOXP3. Now, we made an extract, a methanol extract from Ficus rubiginosa, and we treated cells to differentiate towards Th17 and T regulatory cells. And as you can see here, as I said before, with a mixture of TGF-beta plus interleukin 6. As you can see here, all the methanol extract of Ficus rubiginosa was able to inhibit the differentiation of T regulatory cells, but not the differentiation of Th17 cells. So the methanol extract of Ficus rubiginosa moved the balance between these two kinds of cells, inhibiting T regulatory cells, but highlighting Th17 cells. The first conclusion is just this. Ficus rubiginosa methanol extract inhibits the differentiation of T regulatory cells, but not of Th17 cells. Then what we did, we made an additional fractionation of the methanol extract, and we got the hexane extract and the ethanol extract. And we tested this new extract in the same experimental setting as before. As you can see here, we analyzed T regulatory cells by means of flow cytometry. These cells are defined as CD4 positive, CD25 positive, and FOXP3 positive cells. And as you can see here, again, methanol extract was inhibiting, as we have seen before, and also the hexane extract was inhibiting the development of T regulatory cells. So the important compound for this inhibition are all in the hexane extract, but not in the ethanol extract. Now let me talk a little bit about the importance of interleukin 6 in the balance between Th17 and T regulatory cells. If you add interleukin 6, you favor the development of Th17 cells. But if you take off, withdraw interleukin 6, in this case, T regulatory cells prevail on Th17 cells. This balance is very important in our organism because Th17 cells promote autoimmune aggression, as we have seen in the arthritis, whereas T regulatory cells protect from autoimmune aggression, but T regulatory cells protect also from cancer. So if you want to treat autoimmune disease, you have to shut down, you have to upregulate the number of T regulatory cells, but if you want to protect from cancer, you have to shut down these cells. So what we did is to do the same experiment as before by subtracting, eliminating, withdrawing interleukin-6. And as you can see here, when you take off interleukin-6, you take off also completely the inhibition of T regulatory cells. So Ficus rubiginosa can inhibit still T regulatory cells, but only in presence of interleukin-6. The inhibitory effect of Ficus rubiginosa on development of T regulatory cells is dependent on the presence of interleukin-6. Of course, we tried also to see why there was this connection between Ficus rubiginosa and interleukin-6 by doing a gene expression profile. A lot of genes are modulated by this methanol and hexane extract. Here you can see some of them, for example, LIF, EFT, EFIT, TRIM30D, CD40, CD9, CCL9, sorry. But until now we did not identify the genes that were responsible for this interaction between Ficus rubiginosa and the interleukin-6. So before I leave all of you, I would like to thank my Vietnamese Italian friends that worked on this. For example, Professor Trinity Thuy, she's a professor of chemistry in the Vietnamese Academy of Science and Technology, together with Bati Cham, Dr. Francesco Paolocci at the National Research Council in Italy. And as you see, there is also an intruder among them. Thank for your attention, but before I leave all of you, I would like to show you a small movie, because as someone already said, Pulok said before, in September there will be in Italy the conference, the 25th conference of the International Society of Ethnopharmacology, which I am the current president. And this conference will be in Italy, in Foligno. So all of you are invited, of course. We will have hopefully many people attending from India. And I would like to show a short video about this conference.
Foligno, the beating heart of Umbria and the navel of Italy, is a city with ancient Umbrian and Roman origins, now a vibrant center of commerce and culture. Here, St. Francis of Assisi embarked on his spiritual journey, and in 1472, the first edition of the Divine Comedy was printed. The city stands out for its majestic, noble palaces, witnesses to a glorious past. Palazzo Trinci, a treasure chest of Renaissance frescoes. Palazzo Orfini, the cradle of printing. Palazzo Candioti, the heart of the Quintana. And the Palazzo Comunale, a symbol of civic power. Foligno's spiritual soul is reflected in its churches and oratories. The Cathedral of San Feliciano, the Oratory of the Crucifix, the Church of Santa Maria Infra Portas, and the enchanting Oratory of the Nunziatella, which houses frescoes by Perugino. Twice a year, Foligno transforms into the stage of the Giostra della Quintana, an event that revives Baroque splendor and engages the entire city in an exciting equestrian competition. The cultural scene is enriched by contemporary art, archaeological, and diocesan museums, celebrating the city's rich artistic heritage. Foligno is also synonymous with good living, featuring events like I Primi d'Italia and a lively, dynamic atmosphere along its bustling streets, full of shops and meeting places. The surroundings of Foligno offer breathtaking landscapes: Rasiglia, with its natural springs, the Minotre Valley, with waterfalls and streams, the Abbey of Sassovivo, a place of spirituality, the Hermitage of Pale, immersed in nature, and the Colfiorito Park. Foligno also hosts 2 degree programs from the Faculty of Medicine and Surgery of the University of Perugia, one of the oldest universities in Italy and the world, physiotherapy and nursing. In September 2026, Foligno will welcome the 25th International Congress of the International Society for Ethnopharmacology, titled Ethnopharmacology and Transcultural Medicine. The event, held in the stunning venues of the San Domenico Auditorium and Palazzo Trinci, will focus on the interdisciplinarity and interculturality of ethnopharmacology, a scientific discipline at the crossroads of various fields of knowledge, based on the understanding and respect of traditional cultures. We look forward to welcoming you to Foligno.
Thank you. Let me end just thanking Nishit, which is helping me in these days, and also the driver that brought me last night from Delhi airport to here. And I would like to thank all of you. Thanks.
Thank you, Professor Delfino. We are now open to have some questions. Mike, please.
Good morning. Myself, Dr. Momita Kanjilal. I am from Clinical Research Division. Here I am, you can see. Yeah, it's here.
Okay.
Yeah. Sir, I'd like to ask you one question. Before that, we are very thankful that you have come over here and delivered a nice talk. Sir, in the field of arthritis, what you have presented, it is all the inflammatory arthritis which is shown. Sir, do you have any experience in non-inflammatory arthritis where CPPD crystals are involved, where we can see we can have something for those patients, because for inflammatory arthritis, many treatments are available, but for non-inflammatory arthritis, very less, few treatments are available. I am talking about calcium pyrophosphate phosphate crystals depositions in the joints, which most old age people are involved in that, geriatric cases.
Thank you very much for your question. It's a good question, but unfortunately, we don't have much experience with non-inflammatory arthritis. What I can say is that most probably the decoction of Artocarpus tonkinensis, it works only on inflammatory arthritis because of its action mechanism. As you have seen, the decoction inhibits Th17 cells, which is a feature of inflammatory arthritis. So probably the decoction does not work if there is no inflammation. And maybe you can try the other one, the Ficus rubiginosa, but I don't have any proof about this.
Any other questions from the audience? Please.
Hello, sir. Good morning. My name is Dr. Ram. I am heading the new product development in Patanjali for food and nutraceutical division. So this is a wonderful presentation and most welcome in India. So we have couple of products for this arthritis and anti-inflammatory. So did you check some couple of molecules as well, because this is like 100% herbal concoction? Did you check with like glucosamine or fonditin sulfate or methyl sulfonyl methane, which is very, very safe and treated as in like a nutraceutical and already proven in worldwide? So is any comparison with that as well?
Unfortunately not. I just studied these. It took to me 20 years actually to.
All right. Okay.
Because
Yeah.
you know, with my Vietnamese friend, we started working on this in 2006, and we did some study on leukemia and on arthritis. But it's quite complex because probably you know that the collagen-induced arthritis is a very painful model.
Correct.
I mean, the mice must be very clean, because if they have, for example, a parasite in the intestine, they don't develop the arthritis. So it's a very painful model, and we just checked this compound, not other, but you know.
And this is the collagen, like type 3 collagen, denatured collagen, or how it is?
Sorry?
This collagen, whatever the collagen you have to type 1?
Is type 1 collagen.
Okay.
And you have to, usually any kind of mice can have this kind of disease, but the best one are DBA, DBA1 mice.
Okay.
They develop the arthritis after the challenge with collagen in about 50% of mice, and the other strain of mice, they get lower percentage of disease development. So if you have to use this, I strongly suggest that mice must be very clean and DBA1.
Thank you very much, sir. Thank you very much.
You're welcome.
Good morning, Professor.
Hello.
Sandeep Sinha, nice to see you again. So I just wanted to ask about the first plant, the Vietnamese plant. You said it basically acts very similar to the Th17 monoclonal antibody. Could you please elaborate more on that?
As I said, let me. No.
I can see you, Professor. Yeah.
Yeah, but I want to see you too. So the first row is, So yeah, as I said during the talk, the mechanism of action is really interesting, because it's the mechanism of action of the biological drugs, such as monoclonal, anti-interleukin 17 monoclonal antibody. But this kind of drug, the decoction is better, because if you take a monoclonal antibody, you just target one small part of Th17 cell, the production of interleukin 17, which is important, but it's a limited target. But if you use, for example, a plant like this, you completely eliminate all the cell. So not only interleukin 17, but interleukin 22, interleukin 23. The action is complete. While the action of a monoclonal antibody, which is important, is very good and, put forward the medicine, the field of medicine a lot. But it's not enough, because just blocking one target is good but not enough, and favor also a lot of side effects. While if you eliminate all the cell, you decrease also the side effect and increase the efficacy. This is what I think. And thank you for, welcome me again here in Patanjali.
Thank you, sir.
Thank you, Professor Delfino, sir.
Thank you so much, sir.
Please accompany me in thanking Professor Delfino with a loud clap.
Thank you.
Thank you.
Thank you very much.
I would now like to invite Professor Luis Felix Noties to present a keynote lecture on the topic From Molecular Evidence to Knowledge, AI-Driven Metabolomics and Literature Mining for Unlocking the Chemistry and Medicinal Plant. Professor Noties is a CNRS Junior Group Leader and Professor Chair based at the University Côte d'Azur and the International Institute for Artificial Intelligence, France. Professor Noties is a founding member of Holobiomics Lab, which involved in the development of experimental computational methods for studying the host microbial interactions. May we now invite Professor Noties to kindly initiate the keynote lecture. Let's welcome with huge round of applause.
Okay, 1, 2, 1, 2. Perfect. Thank you. Dear hosts, dear organizers, dear colleagues, dear audience, it's great to be with you today. I'm really happy to discover the research and share ideas. Thank you so much. So I'm leading a research group at the University Côte d'Azur that is composed of both chemists and software engineers. And you will see that in my presentation, we are bridging chemistry and AI to develop new approaches. So there is an issue with the font. I'm sorry for the mess, but I will guide you into the process. Usually, I never start with this slide, which is showing my track record. And as you can see, it's a long way from studying at the university to the PhD degree, the postdoctoral research. It's abroad. It's a very long process. And I wanted here to bring up how it started at the scale of my life. So in 2004, I traveled to India for 3 months when I was 18 years old. And I practiced Kalali Payatu, hosted by a family of Ayurveda practitioners. And I lived with them for several months. And it was one of the best times of my life. And for me, it's very special to be here today. And at the time, I was practicing, so I was doing Panchakarma for the first stage. Then I was practicing every day at 6 a.m. And after a month, I got injured badly. And I couldn't move. And so the grandfather offered me to come to the Ayurveda center to get some care. And he prepared the plaster with the plants. And I was discovering Ayurveda. It was very inspiring. And then I got this plaster. I could see the whole process. And then when I returned to my room, I couldn't move. And yes, there was no internet. There was no cell phone. And the only books that were available were books on Ayurveda in English. And so I started to read for about a week, and it was really inspiring for me to discover this ancient wisdom. And because I have a scientific background from my high school, I was curious about, okay, what are the mechanisms of action? There must be very interesting discoveries to be made out of this. So this is myself. I don't have many pictures of the time, but this is myself, a young man, very excited about science, about the world, and this gave me a lot of, this shifted my perspective about cultural bias and openness on ancient wisdom. And so now back to my scientific career, the research question I'm pushing for is for the concept of the holobiont. So holobiont sounds like holistic. It's the same root, same Latin root, so it's the meaning that it's a continuum of life from the system biology standpoint. And so when we start from the genome, we can get all the way to the phenotype. And one major finding of the last decade is that every single living organism is associated with microorganisms that are part of its functionalities and that can really regulate how it operates. And so the major level for this interaction between the host, the macroscopic host, and the microbiome is the metabolome, small molecules that are like communication messages for it. And so, as I said, all life forms, including humans, are considered as holobiont now. And so if we want to study this organism, we need to combine many different omics approaches all together to have some representation of this living system. And if we add to this the interaction with other species, with the ecosystem, as well at the scale of environment and the scale of time also, with transgenerational effect and evolution. This is really fascinating. And so my specialty is metabolomics. So we don't see the pointer? Okay, let me do this. Up. Laser pointer, up. Yeah. Metabolomics, which is the science, the holistic science of studying metabolites in living systems. So I will discuss more on the progress made for metabolomics. This technique starts from a sample. It can be anything, it can be a plant sample, and we use an instrument, a technique called mass spectrometry, that allows to ionize molecule so we can measure their spectra and get some structural information about their identity. And as you can see, typically we process the data and we start to do some statistical analysis to make sense of it. However, these fingerprints that we are collecting at scale, so it's really fast instrumentation that can collect thousands of spectra in tens of minutes. For many, many years, we were stuck because we had only few reference spectra, about less than 5%. However, the global community contributed by releasing additional reference spectra, enough so we could train deep learning models to improve the accuracy of what we can predict. And so nowadays, these advanced approaches are allowing us to annotate most of the spectra that we can collect. And of course, to make sense of the biological system, if we are restricted to just 5%, it's hard. But with more information, we can start to better propose hypotheses on these systems. So myself, with many colleagues from the US, from Germany, from Europe, we contributed to develop some of the software that are used nowadays to process the data and also to annotate them. So it's a long process to get all this paper together. It's a collaborative international effort. And essentially, we get from the data collected on the instrument down to some information that we can use on the biological system. Okay, this is more problem. Oh, it seems like most of my slides are, we will see. Okay, I will explain. Yes, it's a new language. So this is the pipeline essentially. So starting from the plant, we extract, and you are familiar with this at Patanjali Research. We extract the plant, we perform some mass spectrometry analysis by liquid chromatography. So we separate the constituents and we collect the fragmentation spectra on them. And as I have explained, we can deduce the molecular information molecular formula, the structure, and nowadays, if we don't have the structure, we can have the chemical classes for most of the signals. However, we have these efficient approaches, but it's very complex to interconnect the information. So we have like heterogeneous set of information that we have to aggregate efficiently. And so this is a major challenge. And so how do we make sense of this complex set of information? The first approach is to use prior knowledge. Prior knowledge meaning the scientific literature, or it could be any prior knowledge that could help to understand or to prioritize what to look for. There are also databases that curate information about which structure were found in which plant, for example. But what I wanted to bring up today in particular, this is very important for the rest of my talk, so please bear with me, is the proposition that colleagues made to use what is called a knowledge graph. The knowledge graph here is used to represent this heterogeneous set of information into a continuous representation. And so this is a graph with semantic descriptors on it, meaning we have nodes and edges, and it's describing using natural language the information and their relationship. So for example, we have this molecule that has this bioactivity. It was observed in that spectrum from that given sample back to that species. So it's in interconnecting complex information that are originally disjoint. And the power of this approach that derived from internet, it's from the semantic web, is that there are programming languages, like the Sparkle language, that allows to do data mining on this graph structure. And so, for example, this question about which are the plants that contain most of diterpene esters, we would have the corresponding query that would allow to extract the information. And so when I joined the laboratory of Professor Jean-Luc Wolfender at the University of Geneva, my role was to collect mass spectrometry data on one of the largest plant collection in the world. It's from a French pharmaceutical company called Pierre Fabre. And they have about 17,000 extracts from all around the world. Everything is Nagoya proof. It's a very rigorous collection. And in addition to performing the mass spectrometry data, we run the annotation pipeline. So we generate information at scale and produce this massive knowledge graph from this huge collection of plants. And what happened after we created that amazing resource is that the people that have the insights, they have the good questions to ask. For example, I'm interested in this class of compounds, what are their properties? They are not the same person that knows how to code, to program in the Sparkle language. So there is a gap between relevant questions and how to create the corresponding query. And at the time, ChatGPT was released. And so we started to experiment with large language models if we can automate that process of translating a question into a corresponding Sparkle query. And what we observe is that it does work if we provide enough information. The models, they can hallucinate easily if they are not aware of or they were not trained or not aware of the context. So with the proper engineering, we could see that it was actually possible to achieve that. Furthermore, we thought, okay, maybe the LM could also help, not just in getting the question, but also in helping understanding the results, because not everyone is an expert in metabolomics. So for the rest of my presentation, and I can see that the font is correct, so this is encouraging. It's a presentation in 3 parts. And I will start with MetaBOTI. MetaBOTI is the proof of concept for natural language question translation to a knowledge graph query. And this is a joint collaboration with my colleagues, Professor Wolfender and Dr. Gandon, and with some of my first lab members. We crafted a multi-agent system. So this is really agentic AI. I'm sure many of you have heard of this, are maybe familiar with it. So it's a multi-agent system that operates with multiple specialized agents, AI agent meaning LLM agents, okay? They have instructions, they have a prompt, they have tools, and they are part of a larger system, and they interact together. So the first agent takes the question from the operator and we'll check, okay, is that question relevant? Okay, it's part of the scope, let's continue. Then we have a supervisor agent in the middle that organizes the workflow and that would collect the required information. So for example, we need entities ID, so these are labels for the classes or the species and so on. We need to have a Sparkle agent to run the query. So I will not get into too much detail, but we have this entire system engineered to produce the corresponding Sparkle query, to run it automatically on the server, and to provide a basic answer through the interpreter back to the user. And so how do we evaluate this kind of generative AI system? Of course, we need a benchmark. So we crafted with experts a benchmark of 50 domain-related questions of different complexities and tested 3 different configurations. One, just a ChatGPT LLM with some basic instructions, or multi-agent system, MetaBot, with a medium performance LLM, GPT-4o Mini, and then a higher tier LLM. And in the table I'm presenting now, you will see those 3 configurations, and this column is for the accuracy over 50 questions. What we see is that one single agent is not able to achieve, and this is expected because it doesn't have enough information. However, it's not, I need to speak aloud? Okay. However, okay, wow. However, when we start to have the multi-agent system, we can see that the performance is increasing massively, and up to 80%, a bit more accuracy with the multi-agent system using a very high-quality LLM. So this was encouraging to us, and I will present you after what we are doing now. This is the first time I'm presenting this research. Yeah. So the first subject is, how do we make sense of prior knowledge? How do we make sense of literature, ancient literature possibly, at scale? And LLM has some answer to provide to this. If I take the example of metabolomics, my field, students, they need to be trained in chemistry, informatics, data science, all the way. It's a very long process. So it's challenging. And now that we have larger language models, we can start to imagine other ways to get some training and also to access knowledge. I'm sure some of you are familiar with Perplexity AI, and there are like other commercial solutions to propose a bibliographic search using generative AI. However, these systems, they are black box, first of all. We don't understand exactly how they operate. They are proprietary, commercial, and they are very superficial. They would use the abstract level of information in most cases. There are some full content as well, but usually it's very frustrating. If you are familiar with a given domain, you would try it and you say, oh no, it's not deep enough. So what we proposed is to find inspiration from how us as scientists, we are actually doing the research. We start with bibliographic search. We look at the literature, we curate carefully the content we are reading in those papers, and we are crafting those bibliographic content. Then we can propagate with automated approaches and so on. And also the institutions, they are giving us access to the full content of the article, while with other commercial solutions, this is not the case. And nowadays, a lot of the training is happening through workshops, through code repository. So it's not only in the research article, the knowledge and the know-how is distributed throughout the internet. And what we achieved, we are proposing, is a tool called Perspicacité AI. And the idea is that we want to design an ecosystem for generative AI to do data mining at scale for bibliographic scientific literature. Okay, I can see that my new language is back, but that's okay. We have the symbols. So, Zotero, thank you very much, exactly. So we start from the bibliography, and then we export the file from the bibliography, and then we create a knowledge base. So from all the PDFs, from all the content, we are downloading everything. We assembled a specialized knowledge base on the subject. And that knowledge base is integrated in an agentic AI system. and I will explain this as well. So it's very powerful because you can actually leverage agentic AI on your own bibliographic content that maybe many of you, I know for my thesis, it was a very long process. You already have prepared, aggregated this resource. So you can really leverage it. Also, you have the full text, as I said, and you can also use a local LLM. It doesn't have to be a cloud-based LLM. There are very good local LLM nowadays. So even for sensitive information, this is also possible. Now this is a figure, so all the fonts are looking good. An overview of the system. So we have this knowledge base that is crafted from the bibliography. And then we have a retrieval, augmented generation process. So essentially, when someone asks a question, we look for the resource, for the documents that are the closest in the natural language embedding, and we retrieve those documents. And I will show you on the next slide the different retrieval mode of the agentic system. But my most important message with this approach is that we can track the source of all documents, and we can cite precisely which fact is coming from which research article. So this is very efficient for open and transparent research, and to ensure also that the AI system is grounded in facts and reducing the risk for hallucinations. Now a more advanced view of the system, and I want to comment on this mode, a profound mode, which starts with a planning step. So we are decomposing the question in sub-questions, and the system will retrieve documents for the first step and evaluate if the documents are relevant enough and it's sufficiently answering. If yes, it will continue with step 2. If no, it will continue to search for information. At the end, we are producing a report, and this report has a very comprehensive summary with all the references that are included. So as you can understand, this is useful for training, and I think there are opportunities to create new courses, interdisciplinary courses in particular. More importantly, it's going to be useful for AI system to do large-scale literature survey to generate new hypotheses in a semi-automated manner. And now I'm heading to the last part of my talk, and I still have 15 minutes, so I will just slow down a little. This is, to me, this is the future of research. The first system I have presented is a rigid multi-agent system. So we have crafted an entire architecture. We have defined all the agents, all their prompts through iteration and so on. However, if we would have to modify it, we would have to re-engineer it, and it's not sustainable. So the future is to have some more adaptive AI system, and this is very much in line with the process of scientific research, because we don't know what we will be discovering in this process. It's an open-ended process. So our AI systems, they need to be able to pivot and to decide, okay, this hypothesis was not relevant. I need to modify how I operate conceptually to produce this. And so, indeed, we need to have this AI system able to fully adapt, or what we call self-evolved. They need to, like a synthetic living entity, they need to adjust to the tasks that are asked. So this is needed, but this is very challenging to achieve that. Okay, some of the text is not shown, but that's fine. So let me present you Mimosa AI, and the quality is low resolution. I'm sorry for that. I will explain you. So essentially, it's a multi-agent system, but it's a self-evolving multi-agent system. You will be given, for example, a scientific task to the system. The system will decide, okay, to achieve this metabolomics pipeline, I need to create this workflow. And I will try, as a first attempt, to have this multi-agent, these agents cooperating together. And these agents, they have a set of tools. So they have access to, for example, lab instruments, any programmatic interface, Python libraries, or any bioinformatics software can be made available to these agents. And so the workflow will be crafted in order to try to achieve the task. And then we have a process where we execute the task in a totally autonomous manner, And we have a judge that will look at the results and say, okay, no, this is not correct. We need to change completely. We need to bring new agents. We need to modify that particular agent and so on. And this is the process that we call the mutation of the workflow. So we are going to do one single change at a time, either on the agent or either on the multi-agent system architecture or organization. And whenever the judge estimates that this is good enough, we will save the workflow for future use, and we will use this as a starting point for future research. So how do we study such a system that is really, I would say, not wild, but a bit hard to predict the behavior? It turns out that there is a benchmark called Science Agent Benchmark that has about 100 tasks from scientific literature in different disciplines. And so we evaluated the Mimosa AI system in 3 configurations. So the first configuration is a single agent. So it's Mimosa AI, but a single agent is operating. Then we are starting with the one-shot multi-agent. So it's the first workflow that was proposed by the orchestrator. And then the meta-learning, which is a process of reflectively looking at the first workflow and the results and deciding to modify it, to mutate it in some way. And so the interesting results that I'm showing here is that a single agent with a high performance reasoning model like DeepSeek v3.2 is reaching more than 38% of these 100 of tasks. If we start to bring multi-agent one-shots, the performance are actually decreasing. And if we bring in the learning phase, then it's increasing again. So this is showing that the meta-learning process is helping the AI system to actually improve itself to some extent. At the same time, the fact that a single agent can already achieve a lot, it shows that sometimes it can be beneficial to have a single agent because the memory is shared in a different manner than with a multi-agent system where memory management is challenging because you have this context window with LLM, so you don't want to destroy the window. It's very costly and it's losing efficiency. So there are like improvements for sure on this side. So you can see that today, on the benchmark of scientific tasks, AI system could do almost half of them correctly as of today. And there are other groups that are proposing other approaches, similar order of performance. So this is going to transform radically how we do research in a semi-automated manner. So preprint, not yet available. In the coming weeks, I'm working on it now. But I wanted to bring that example. So this is a research article from my PhD thesis. So I'm a chemist in natural products. So I worked on plants from Corsica. They are very complex diterpenoid structures, difficult to isolate and to characterize. This was a lot of fun. And these compounds, they have antiviral properties. And so in this paper, we are proposing a method to look for the bioactive compounds in the molecular networks. So this is a molecular network here. To propose which are the bioactive compounds from the bioassay guided fractionation using a statistical approach. And so this paper was really well received by the community and very much cited. And so what we did is we gave that paper to the AI system I've presented you. And we gave that the PDF and said, okay, reproduce the analysis that was done. And at the time, I have to say, we provided all the data sets. So all the data were available. All the code was on GitHub. So everything was there to reproduce the research. Otherwise, it's a different story, right? It needs to be like reproducible research. Otherwise, there is no point. And so what happened is this. I have this short video to show you the process. Everything is programmatically done, right? It's like terminal command line tool. But this is a visualization frame. And you have the summary of the first step the plan, the workflow was created, and then it's entering different stages, the time it took, the tokens, and so on. So this is the video. Just it's about 25 minutes in video not showing. No, it's slow. Okay, let's return. I will try again, and if not, we just continue. Please. So you see, the AI system goes into each step. This is a time lapse, about 25 minutes. And so it goes into each step and try some code, make some reasoning on it. Is it failing? No? Yes? Okay, I continue, I try something else. And it goes all the way into that iterative process of self-evolving, trying to achieve the tasks one step at a time. And as I said, it took about 25 minutes. In my PhD work, there was more than this, but this part was 2 months of work by at least 3 people, full-time. And this was done in less than half an hour without any other instructions than a PDF article. Okay, so I made a nice drawing. It doesn't show well. I will use different approach next time. But essentially, I wanted to have some concepts or philosophy on what is the future of science. And today, it's clear that with we are able to bridge many different parts of research that were disconnected or hard to connect. Of course, we have the prior knowledge. We can do experiments at scale. So with metabolomics, we can run thousands of analyses, and we can also generate hypotheses. So systems like the one I have presented, they will be able to leverage all of that in a semi-automated manner because they can access the scientific literature. They can process the experimental data. They can propose hypotheses because they have the literature, they have the data. So they can just propose something. And in the future, as I said, Mimosa AI, it can control some instruments. So we have a mass spectrometer that has a programmatic interface. We have a pipetting robot that can be controlled with Python. And so those could be made available to the AI system to decide, okay, this hypothesis is interesting. I need to craft a new experiment. So let's proceed with that experiment. Let's collect the data, and so on. So in the future, all of that will be integrated with infrastructure and AI system at scale. So clearly, that will bring opportunities for many novel discoveries. And with novel discoveries, we will have also newer instrumentation that will allow us to discover, to observe more of the natural world and better understand health. So as I said, and I wanted to make this connection at the end of my talk, prior knowledge in the sense of science classically would be the scientific literature. But what prevents us from bringing ancient wisdom in that process? And I would like to propose something else, something even more integrated. What is preventing us from having the AI system, the self-evolving AI system, being inspired by some philosophies or some concepts that are related to a culture? Why not hybridizing different approaches in how the AI system and maybe screening for different self-evolving AI system with different philosophy or different way to organize and address scientific questions? There are many opportunities, I think, to imagine future tools for the research. So I wanted to acknowledge first my research group. I'm very lucky. I have very talented colleagues. that I work on every day. So thank you so much for them. They are the one implementing the research vision. And of course, I want to acknowledge funding agencies. So it's mostly the French government and the University Côte d'Azur. And I have also colleagues that are involved in this project at many scales. And yeah, I wanted to say merci and Bahut Dhanyavad. Thank you very much.
Thank you very much, Professor Nothias, for the exciting lecture. Now, the floor is open for the questions. If anyone has questions, please raise your hand.
Louis Félix, thank you very much for this very inspiring lecture, and which really showed us the fascinating future using AI. And I think we could discuss this lecture probably for an hour. I have three short questions. One is on Mimosa. Who is the judge? Is the judge a human being or is the judge AI?
Yes, thank you very much for your question. And I will just bring that slide. In that system has only AI agents as part of it. So the judge would be an LLM, a large language model, that would be charged of judging the performances. However, and this is why your question is hitting a very important, a crucial point, is that we need to use a different LLM. If we use the same LLM, it tends to self-encourage, or it goes in the same direction as the LLM that was used in that process. So there is a need to combine different large language models. Some of them were trained on different languages, on different data sets. So this is important to keep some elevation on what's happening.
And the second question is more chemically related. All this data you presented, or at least which are nowadays mostly used, are MS data and MS spectra. And we all know that mass spectrometry has some shortcomings with configuration of double bonds and also with chirality. And on the other side, we also know that the configuration and chirality plays a very important role for activity. So do you see any chance in the future to include this information somehow? Because I think it's necessary to develop further. Currently, we are lacking a lot of important information with respect to activity.
Yes, you are absolutely right. This is a major, I mean, this is a limitation of mass spectrometry. We can propose some structural properties, let's say the 2D, the planar structure, but the full structural elucidation needs orthogonal techniques like NMR or others. So we are limited. However, the benefit is coming from the scale. We can do this at massive scale. And so I believe the way to see this is that it's for the first stage of discovery, to observe some patterns. And then it needs some follow-up experiments that requires the isolation of the molecule and their characterization. But there are also, maybe in the future, there will be like orthogonal techniques included in the analytical part, like ion mobility in a better manner, or infrared, I don't know.
Hopefully.
Yeah, it would be nice.
Thank you.
Anyone?
Louis, thank you very much. That was a fantastic talk. I mean, I was very happy to see the applications of AI into this segment. And I had just one query. I'm a biologist, so I see this with the eye of a biologist. So when you talk about metabolomics and seeing the changes that's happening, but if you bring in a live cell, I mean, when the compound goes in a biological environment, how are those changes going to happen? Can we ask AI or the Mimosa AI to help into it, or predict that this may be a potential integration of these chemical structures with the biological function.
We would have to try and look into more details, but what I can say is that if the analysis can be reproduced programmatically with informatics code, then there is no reason why such a system couldn't reproduce it. Especially if we are bringing some prior knowledge about what is the good practice to process the data, what's the background about the discoveries of this way or whatever mechanism that would be studied. So I think it's just a matter of engineering or providing enough information, and eventually this kind of analysis, they would be streamlined and automated to some extent.
I think that's actually very nice, and I really thought it would come to the surface very soon. But my thought was slightly more focused on that you have a very nice integrated stuff for the chemistry of the compounds, and then you have a very nice biological pathway information in public domain. But somehow the integration becomes in concerns when you go to take them together. So my thought was that can we have an AI as the glue to connect both of them and then find something which is already not known to us. I mean, it's like some sort of a novel pathway analysis or some sort of changes at the level of the compounds, but that which might drive the biology behind the functionality that we see clinically or preclinically.
Yeah, that would be very much needed. And I see the community is pushing toward that. It's not there yet, but for example, there was a paper recently released by Dorinchin Group where they use generative AI to annotate compounds, because many compounds, they are not in databases. We don't know their pathway, for example.
Right.
We can deduce it as an expert. But it's possible to use large language model to look at the literature, the information on these compounds, and classify them in an automated manner. So there will be opportunities to just provide more information, and in the end, and this is where I think, you know, it's completely transforming how we will do research in the future. So what is the role of human researcher in this? I don't have the answer to that. I just know that technically this is possible, and it will necessarily happen because it's very cheap and it's scalable. And so the role and the place of researchers might be to be as a guide to the orchestra, to provide all the insight you have to the AI. Absolutely. So it's going in the right direction. And
absolutely, I really like that connections. I hope the orchestra plays very well and the music keep on going. Thank you very much indeed. It was a really nice presentation. Thank you.
Anyone from the audience?
So thank you very much for this great presentation, and I think this is a very nice tool, Mimosa AI, because if you fail early, it always saves a lot of time and effort as well. So I'm talking about my almost 15 years back, I was working in the medicinal chemistry, and there is a very famous plant in India that is called curry leaf, and curry leaf has a lot of potential for hair growth, etc. So in my last company, we isolate couple of compounds, we do a lot of biological activity for the hair growth. And once we do the NMR and all things, and we finally found after 2 years that this compound is already known in the library and patented by someone else, like carnitine oxide A and B. So I think your tool is very nice, and is free, accessible to everybody. It's really helpful for the scientists and scientific community. Thank you very much for this. That's my input.
Thank you very much. Thank you.
Professor Luis, it's an excellent presentation, no doubt about that. In your research also very in-depth research. One important thing, can you highlight about that? Why when you are incorporating AI in your research or any clinical research, if something goes wrong like that, it happens because AI is not in perfect totally. So who will be responsible for that? And any ethical points of view in your system?
I have no answer to that. It's a clear challenge, and it's necessary to have much transparency in the AI process. What I can say is that this system is recording every step it's performing. So we have a log of every single interaction, and so if there is an issue, we can track where it happened, and so in the end, it might be more transparent than how a human organization might operate, or human brain maybe might operate. But for clinical purpose, then this is very, very delicate. This is really, the community is at the stage of prototyping. But maybe in a few years there will be some efforts for clinical.
That's true. How will you get the ethical clearance? Is it easy to get the ethical clearance to that when you're incorporating AI into your research?
I have no idea. Yeah.
Okay, thank you.
So, thank you for the wonderful talk. I have two queries actually. Number one is, what is the flexibility of this system? Can you also work with metals and see how they are affecting the metabolomics activities?
Yes, more likely. I would say one would provide, the prompt of the system is really what guides the tasks that are decided. So if someone is asking your suggestion of optimize the process, it might look for different processing software, different annotation software.
Okay, yeah. Because we in the Ayurveda have a huge library of something what we call the metal-based bhasmas, and they might be affecting the this metabolomics, and that type is we can correlate with the metabolomic data, right? The second thing is we at Patanjali Institute, under the blessing of Param Puja Acharya Ji, have a huge domain of information in the third type of World Herbal Encyclopedia, which is actually a library of different type of phytochemicals and how they are affecting different type of diseases. It's of course under the supervision of Dr. Ved Priyaria as well. So is there any way that to even further advance this type of database, we can incorporate the metabolomics and then dig into further how to enrich the data from this classical medicine like Ayurveda and see what are the key points affecting a certain type of metabolomics in a certain type of disease?
Thank you very much for your comments and question. I think there is an enormous opportunity to interconnect large literature, encyclopedic literature, with massive metabolomics analysis, because one could look for compounds and try to do some matching with the properties. And it's so much information that an AI system will help to perform some scalable hypothesis search. So certainly, I think in the near future, this will happen. This is technically already possible, and it's going to be it's going to accelerate knowledge generation, especially for deep knowledge that is ancient knowledge carried throughout the generation. And so there is an opportunity to build on this for new AI tools on this foundation. Clearly, I'm convinced by this.
Thank you. Let's give a big hand for Professor Nothias.
I would like to request Param Shraddhe Acharya Ji to please come to the dais for felicitating Professor Meenu Singh Ji from All India Institute of Medical Sciences, Rishikesh, and her husband, Professor Rajendra Singh Ji from PGI, Chandigarh. Professor Singh, sir, was previously has visited the institute 1 year back, and it was a very good surprise for us that he is visiting us again. So thank you very much. Now, may I invite Professor Meenu Singh ma'am to kindly present your keynote lecture on the topic, Integrative Medicine, Bridging Tradition with Modern Medicine. Professor Singh is an eminent academician and clinician and a healthcare leader with over 35 years of distinguished service in pediatrics, pediatric pulmonology, telemedicine, and evidence-based medicine. She is currently serving as the Executive Director and CEO of All India Institute of Medical Sciences, Rishikesh, where her visionary leadership has led to patient-centric and care, innovations and academic excellence. Professor Singh has also led the pediatric pulmonology and telemedicine at PGMIR Chandigarh and holds several professional fellowships. And she has been awarded with several prestigious national and international awards, such as Bharat Ratna Dr. Abul Kalam Gold Medals. So I request ma'am to kindly go ahead with your presentation. Thank you, ma'am.
Thank you, sir. Honorable Acharya ji, Dr. Baike Gupta, jo baithe, and our honorable guests who've come from abroad, and the faculty from Patanjali Yog Peeth, as well as all the students who are there. I was just wondering how my talk will be landing after a highly technical talk on artificial intelligence, which has just been delivered. My talk is rather simplistic, and it is more about how we are going to integrate our traditional medicine with modern science. And as you know, we are at the moment living in this age of integrative medicine. And actually, whenever I come to Patanjali, I always feel that, you know, there is a river of spirituality which is flowing in this place. And whatever you start talking, you attach a bit of spirituality to it. And I was just wondering that in this modern era, when we are talking of molecules and, they're getting affected, and all the photons and the quantum theory which is coming up, how are we going to handle spirituality and the mental health, which is associated with our modern day living and the stress of life, which has always been there. So the title of the talk is, of course, Integrative Medicine and Bridging Tradition with Modern Science. And we know that our ancient science of Ayurveda is reft with so many molecules, so many medicines, which could even be called as drugs, so many systems which have been used for betterment of life, such as yoga. And all these have been there and running for centuries and incorporated in our lives, so much so that an Indian kitchen actually contains the kind of spices it contains. They are actually almost like medicines, they are drugs. For example, we have turmeric, which gives us curcumin. We have, this pepper, which gives us, say, piper longum, which gives us piperine. There are so many agents which are helpful in health and are presently being used in so many diseases. And along with yoga, we are able to modify our lifestyle. In fact, it's science which tells us how to live. And that is which relieves us of our stress. And all these things combined could actually lead to what we want to achieve, and that is longevity. That means, and that longevity is something which is being studied in different formats, and something like telomeric length of our chromosomes, how they're getting modified, epigenetics, which is like transmitting our environmental influences into our genes and those getting passed into generations. So all that ancient knowledge was already existing, and that is what, was always being taught to mothers, what they should be thinking about, what they should be eating about when they are carrying a baby, and how that is going to affect the life of the offspring which is going to be there. So So we know that there are different routes to God and there are different routes to health. And health actually can be achieved by a very healthy lifestyle. That is, of course, number one. It is something which is also endowed in your genes. We know that whatever we might do, sometimes our lifespan is always moderated by our genes. And, we were actually talking to one senior professor and who said that, you see a horse who's working all the time and who's running all the time, but his lifespan is, say, a given number of years. But then there is a python who's, lying down all the time and is not moving so much, but will actually live up to 1,000 years. So it is actually in our genes that it is ingrained how much a person is going to live. But for a given person or for a given species, there is a certain lifespan which has been recommended or which has been outlined, and say for humans it is considered to be 100 years, and that is why you have those 4 phases of life, which starts with your childhood, your adulthood, when you become, Vanprasthashram, and then you have Sannyas ashram. So all those things have been divided into these 25 years of lifespan. And in every 25 years, there is a recommendation of how you're going to lead that particular span. For example, in your initial years, you're going to indulge in acquisition of knowledge. You are also going to indulge in acquisition of good health. That means sports and physical activity. Your learning of cultural values. Those are the things which would come in the first years. And subsequently, because you have learned all that, it will be translated into your adult life, which means that this is the time of reproductive phase in which you're going to have children and you're going to grow them up, as well as you are going to have some professional life in which you are going to build something for the society and for the nation. And everything, as we know, if you follow the principles of Bhagavad Gita, it is always that you are not doing it for yourself. It is something which you are doing for the sake of the world, for the sake of the society, and you are not expecting anything in return. And subsequently, you go on a spiritual sojourn for reaching the higher values and getting what we call as salvation. So another concept which has emerged these years, and everybody is talking about it, including the World Health Organization, is that of One Health. And this is what we also call as Vasudev Kutumbakam. We have been discussing this concept that the whole earth is one. And everything which is on the earth is also one. That means we grow plants, we have animals, we have human beings, we have our total ecosystem, we have our environment, and that constitutes the earth has to be, we have to, because we are all inhabitants of the earth and humans are expected to be the most evolved of the species, they have this responsibility of preserving whatever's ever been given to us by the divinity or given to us by nature. And as we can see that there would be animals which are there are humans, there are plants, and they all have diseases. And they have their own health which has to be looked after by the humans. And that is the reason why we have to worry about our animals and we have to worry about our plants. And these animals are not just the animals which are giving something to us, for example, cattle, or, because we get, say, milk from cows, we get milk from buffaloes, but these are not the only one we have to worry about. We have to worry about all the wild animals, even the stray dogs, because they can all carry diseases, and those diseases can affect us. And at the moment, we don't even know that probably it was a zoonosis which came to us in the form of COVID, and there were some carriers which were there, which we missed, and subsequently there was a huge pandemic in the world. So after that, people have really started thinking about One Health. We have started worrying about plants. We know that if a disease is there in a plant, and if we consume that plant, there is a possibility of we acquiring that particular disease. For example, there are many plants, if they are infected with fungus and, say, aspergillus, there's a possibility that if we consume that plant, there is you can get a disease. For example, sometimes pulmonary disease, sometimes digestive disease, and sometimes even cancers. In some of the viral infections, if they infest the vegetable world, those can become, they can have oncogenes, and they can, or they can initiate oncogenes, and that can lead to cancer process. So many of these things are important, and we have to worry about our plants, and that is what we are talking today, that plants which give us drugs, which give us medicines, also have to be looked after, because if they become diseased, they are also capable of transmitting that disease to humans. And same is true of animals, and about animals we all know that there are many diseases which come to us from pigs, diseases which come to us from mosquitoes, from sandflies. So all these have to be controlled. If we control our environment, only then we will be able to survive ourselves. So now, can joining of paths lead to greater achievement? Yes, of course. And that is the main principle of integrative medicine. We know that Western medicine, which we all learned, which is based on a very analytic approach in which we try to be evidence-based. We call ourselves evidence-based because whatever research has been conducted, we try to critically appraise it and then take the results of that research after critical appraisal into application of practice. That means we take it into practice. Whatever we use in our patients, that is something which is coming out of research findings, and those research findings have already been critically appraised. But there are many other systems of medicine. For example, we have a whole department of Ayush. We have Ayurveda, we have Yoga, we have Unani, we have homeopathy. We have many other systems like Tibetan systems of medicine, Chinese system of medicine, which have their own theories, and they have their own set of drugs or medications or systems. For example, acupuncture, which comes from China and is very helpful in pain relief and in many other conditions. So can we integrate many of these systems? And if we have integration of systems, for example, we've recently started in AIIMS Rishikesh, a department of integrative medicine, in which our allopathic doctors who have been trained in the Western system of medicine are actually trying to work with the graduates of traditional systems of medicine, and they are trying to devise new treatments or new therapies which are very, very effective and which actually prolong the relief which otherwise is not available for one single system. So of course there is a need for studying the outcomes, and there is a need for evidence-based medicine. And evidence-based medicine is something which we take it lightly. I mean, it is a term which has become very, very common these days, and everyone is using it, that our practice should be evidence-based. But it is not easily done. It is something in which you would have, you need to have research findings. For example, you will have sometimes some observational studies. You will have sometimes which are interventional studies. In the interventional studies, you will find that you are giving an active principle to one group of patients or subjects, and you're giving a placebo to the other group of subjects, and then you're trying to do certain trials, which could be randomized controlled trials. And there is a whole pyramid of evidence-based medicine in which personal opinion, which, say I'm trying to give right now, is at the lowest ebb. And at the top is systematic review or meta-analysis. And this systematic review and meta-analysis is something which is conducted on existing clinical trials or existing studies if they are observational in nature. And there are definite tools of searching literature. And now with the availability of artificial intelligence, we are able to do that with much greater ease, which otherwise we used to spend months together in conducting systematic reviews. It has become much easier now with the help of AI tools that we are able to search literature in a very systematic way easily, and then even critically appraise many of these studies in which we need to use what is known as risk of bias. And that risk of bias can easily be conducted by using certain softwares which are now available, and with the help of, that also has been created with the help of AI. And we can conduct our systematic reviews in a very fast way, for example, say in a couple of weeks. So that result which comes out of a systematic review and meta-analysis is something which is going to improve or going to deteriorate the outcomes which we are expecting. And these outcomes are, say, disease outcomes. For example, for hypertension, it could be control of blood pressure. For diabetes, it could be control of, say, fasting sugar, or it could be HbA1c levels. So those are the clinical outcomes which are kept in mind, and along with that, there are certain outcomes which are which are based on quality of life. And if a certain intervention is improving quality of life, that obviously is going to be the best one in comparison to the one which does not improve the quality of life. So we need to have proper outcomes for studying a particular intervention. Now, some systems can be good for holistic health. So this concept of holistic health is something which has now come in after integrative medicine, that we are trying to achieve the best quality of life, a health which is in all spheres of our activity. For example, health as such was defined as, it is a state of physical, mental, and spiritual well-being, and not just absence of disease. So, and that is what is actually holistic health. It is something which actually improves your outcomes right from the word go and right from your inside, from your core. And that is why some systems are very good for holistic health. And of course, we have yoga at the top of it, and Patanjali Yoga Peeth has actually popularized yoga all over the world in such an effective way that the whole world now looks towards this institution. Then, of course, there is meditation, for which there are many ways to learn it, and there are many institutions which might even teach students how to do meditation, dhyan, which I think all of you who are studying in Patanjali are learning. Then there is reflection, that is to think about what we are going to do. It is like looking inside and trying to reach an evolved state of being. And everything has to be balanced, and our thought and our actions have to be balanced. Something which comes in our thought has to be delivered into an action. And of course, there are other features which would help. One is closeness to nature, pure diet, and conserving the environment. These are some of the features which are very important for holistic health. Now, some people may debate whether amalgamation of Ayurveda, homeopathy, Unani, and allopathy, is it totally possible? And we have always been thinking that all systems of medicine ultimately are delivering in the form of some systems, or they have some medications, and those medications are derived mostly from plants, but they can also be derived from other elements, and say from bhasma, which are derived from salts, which are inorganic, or sometimes organic products, other organic products which may be there. And some people may say that it is not possible to combine many of them. And actually, one can think about this and study these various systems of medicine. For example, Unani, they also have medications which are derived from plants. Homeopathy also has medications or extracts which are derived from plants. Similarly, Ayurveda, of course, is entirely based on plants, and so is also Siddha, which is considered to be an offshoot of Ayurveda, which is practiced in the south, and is based so much on the derivatives of plants. So the study of structure and function is central to all components, but the mode and constitution of therapeutic approaches may lead to some confusion. And that is why there are certain things which are not natural products, which may give rise to drugs, foods, which will be in the form of nutrition. Then there are certain psychological interventions which are helpful, for example, in yoga and meditation. And mind-body practices is something which is now becoming a very important part of clinical medicine. Actually, there are centers for mind-body medicine. Of course, they were always there in Uttarakhand. For example, in Rishikesh, we had so many centers which are telling people how to practice mind-body medicine. And hence, this demarcation between psychological and physical has to be defined. For example, physical ailments which cannot otherwise be cured, of course, they have to be treated with devices and surgery. So this is one small proportion in which you would need an active intervention of surgeons or of interventionists, but many of other diseases or ailments could be cured with the help of drugs, and many of these drugs would be derived from animals. So there was an ethno-medical investigation of medicinal plants of Chakratha region in Uttarakhand, which are used in traditional medicines for diabetes by Jaunsari tribe. And many of these medications are, as you can see, this is a map of India, this is Uttarakhand, and this is Dehradun, and somewhere here is Chakratha, and this is the region where this study was conducted in, and they tried to take many of these plants. And actually, if you happen to go to Selakoi, which is a place close to Dehradun, and near Ponta Sahib, this where we have a center for aromatic plants. And they are not just working on aromatic plants, they are working on many medicinal plants which produce oils and which produce aromatic products. And these products are really helpful in treatment of diseases. And in diabetes, they had, this is a schematic diagram showing anti-diabetic mechanisms of selected surveyed plants and their bioactives. And there are many of them, and we know that there are many drugs which are available to us in the allopathy, and there are many medicines which are available to us in Ayurveda. And Patanjali has actually contributed so much. And right now somebody was talking about Karela. And from Karela there is a very effective medicine, I think, which has been discovered and is being used. And really thankful to Dr. Varshne, who is working in the Patanjali Research Center, because that particular medication, it appears, can replace insulin. It's like an oral insulin, which is there, and it must be tried in the form of a clinical trial so that we can recommend it to in a larger section. So plants to medicines is not a new concept. We know that many plants as a whole or as various components of plants, for example, their leaves, flowers, stems, or roots are used in the form of medications, and no other place than Patanjali to learn about all these medications. They have Patanjali Pharmacopoeia, in which all these drugs, and I was just told by Acharya ji that it has been converted into a database, into a website, in which you can actually find all information about all the medicinal plants. What is, their botanical name, what is their general name, what is the component which is given by them, and those can be tried. And I think it is a wealth of information in which Indian medicine can actually go forward. And even Indian pharmacology, Indian pharmacopoeia can go much forward by discovering these new medications which will come out of these plants, which are so far unknown to the Western world. So we know that plants as a source of all medications. The Ayurveda derives many therapies from herbs. Homeopathy also have plant components used in therapy, and so are Unani. Siddha seems to be an offshoot of Ayurveda, as I said earlier, and many modern drugs used in allopathy have been derived from plants, and many of them are actually anti-cancer drugs, anti-asthma drugs, even from rheumatological problems. And recently there has been a greater use of CBD, cannabinoids which are derived from cannabis. They have also are being used for pain relief, and they have been also found to be useful in prolonging the life of individuals. So naturally occurring molecules which are derived from plants can act as templates for several new drugs to be used for diseases where there are no satisfactory cures. And when listening to the talk on artificial intelligence, we know that these are the templates on which new drugs can be designed and can possibly contribute to the betterment of human civilization. So with those words, I thank all of you to give me a patient listening. Thank you very much. Jai Hind, Jai Bharat. I can take up questions if there are any.
Audience, please, questions.
Ma'am, I have a query. My name is Anurag. So, ma'am, you talked very well about botanicals, plant-derived components, and I think one of the biggest need, as it seems like, is to generate the clinical evidence which is acceptable across the world. So, there have been a lot of movements and thoughts about doing clinical trials on those formulations. But sometimes there is some sort of hesitations from modern practitioners who said if you have to take bhasmas, they don't want to take it forward, and if it is a very complex formulation, there are some degree of hesitations too. So ma'am, how do you see it, and what is the way forward? How do we can take it forward and convince our modern practitioners to conduct those dedicated, randomized, placebo-controlled trials of the formulations which are complex and also contain bhasmas?
Thank you very much. This is a kind of question which is very close to my heart as well. I was looking after a Center for Evidence-Based Medicine at PGI Chandigarh, which was ICMR sponsored. And we were actually conducting clinical trials as well as critically appraising clinical trials which had already been done. And we do find that there is a dearth of clinical trials in our traditional systems of medicine. As you have said, that there is a hesitation on the part of clinicians to take up, these formulations for clinical trials. But I don't think now with the oncoming of evidence-based Ayush, and Ayush is actually recommending so many trials. And we at AIIMS Rishikesh are conducting trials in the use of Ayurveda and yoga for, say, reduction of sarcopenia in old age. We are a center for geriatrics where we are doing these trials. So I think now the trend is changing, and even the funding agencies are funding these trials in which traditional medicines will be compared to, say, allopathic medicines, and one can actually see the result in the form of pre-derived outcomes which will be there. So I think now it is, and we would certainly invite you to come and participate with us in some of these clinical trials, and the whole AIIMS network. Recently there was the start of this, it's called PARC, it is Pan AIIMS Consortium for Research. And what was found there was that we can conduct clinical trials in that particular network, and special emphasis on Ayush and on Ayurveda, in which traditional therapies could be used in the form of clinical trials. And there are many diseases in which you may have lesser sample at one center. So for doing these multicentric trials also, because we have this network of 22 AIIMS, that we can conduct clinical trials on large variety of diseases, and predominantly at present on non-communicable diseases, because that is something which is coming as a third epidemic that we have right now, and also for cancer, which would be part of non-communicable diseases. So I think all the drugs which you have discovered, I would say, and you have published their structure, right. And I can see so many of them published, and all the publications which have been there where you have published in high impact factor journals. I think they can be adopted and they can be tested in the form of trials.
Thank you very much, ma'am. Thank you
Yeah.
for this assurance.
Yeah.
We look forward to work together and start generating those body of evidence.
Yes,
which would be
that
acceptable.
evidence, then when there are multiple trials of one single agent, one can actually pool them up and then see what is happening. And in the form of multicentric trials, maybe we can have a large enough samples, even without pooling.
Sure.
Of,
Thank you very much, ma'am.
data.
Thank you.
Yes, sir.
So any more questions?
Good afternoon, everybody. Thank you so much, madam, for your talk, and we really enjoyed the talk. I just wanted to bring to you information that we had one clinical trial along with a PG student with the AIIMS Rishikesh last year, and just 2 days back it has been adjusted the best thesis in the whole of India.
Okay, thank you so much.
Yeah, yeah.
I think
The
that
one
is
from Dr. And the second trial is ongoing now.
Okay.
With the geriatrics department with Meenakshi Dhar.
Okay.
And we are looking for more trials from your side, madam.
Actually,
A
there are
combined
many
combined
trials, and recently we had this, there is a team which has come from USA, and there is a, on the, just like Dana-Farber, they have this Institute of Cancer Research, and
Okay.
where even at the level of cell, cell level,
Okay.
there is testing of certain molecules which can happen. And
Okay.
actually, I have visited the Patanjali Research Institute, which is there, and they have wonderful facilities for some of these testing of molecules at cellular level.
Okay.
And many of these other agents are also there, for example, for CRISPR technology. Now
Right,
they are
right,
they're using
right.
a large number of technologies, and So we are certainly collaborating with them, and we are of course collaborating with you. I'm very happy to know that one of our researchers has done that study.
Yeah.
Thank you very much.
Thank you so much. Thank you so much.
Thank you very much.
Ma'am, namaskar. My name is Jay. Actually, you mentioned cannabis.
Yes.
So actually, in India, beside the seed, it is not legalized. You mentioned CAP is working on this mission. Mission cannabis. But is there any scope in near future that it will be legalized in India?
Actually, what I found, say in Rishikesh, because cannabis is supposed to be Bhagwan Shankar's, favorite. People use it, and many people who are in psychiatry, in addiction psychiatry, they also say that it is less addictive than many of the other addiction substances like alcohol. And cannabis is a huge variety of species. Not all of them are addictive. And some of them give you a very good pain relief. And actually you should get in touch with this Center for Aromatic Plants, which is there in Selakwe. And those are the people who are actually working on it. And they are creating a library of genetic makeup of some of these plants. And those plants will then become available. As we know that many of these drugs are derived from cannabis and they are used clinically because they are required for pain relief, for inducing anesthesia or inducing analgesia. So there is a body of, I think, researchers now who are coming out with strong evidence, and especially with the help of nanobiotechnology. Some of these molecules are really being used in cancer care, in palliative care. And so I think this, our own group of intelligentsia, they have to create a body of evidence so that we can impress upon the drug controller and government of India to at least, permit some kind of use of this particular molecule and encourage more research on this.
Thank you.
Yeah.
So thank you very much. I would request everybody to kindly thank Professor Meenu Singh ji for the excellent talk with a loud clap. At the conclusion of the first half of today's session, we would now like to request Param Shraddhe Acharya ji to kindly come to the dais and present mementos to our esteemed speakers as a token of appreciation. So I start with Professor Y.K. Gupta ji. Professor Pulok K. Mukherjee ji. Professor Domenico Vittorio Delfino. Professor Luis Felix Nothias ji. And Professor Meenu Singh ji.
हमारा सौभाग्य है कि हमारे बीच में वारी सोलर बहुत बड़ा ग्रुप है, उसके चेयरमैन साहब हमारे हितेश वारी जी और उनकी धर्मपत्नी जी दोनों बैठे हैं, दोषी जी। मैं निवेदन करता हूँ मंच पर आ जाएं एक बार, थोड़ा सा बस आ जाओ ऊपर आ जाओ, शक्ल तो दिखा दो, फिर अपन चलेंगे। एक बार आप जोरदार करतल ध्वनि से उनका स्वागत करें।
With this, we have successfully completed the first half day of the day one session. We would now request the chaperones to usher their guests to the food launch. I would also request respected professor, faculty members, students, Patanjali University, Ayurveda College, BNY's members and Patanjali Research Foundation to kindly proceed to the university cafeteria for the lunch. I request everyone to kindly return back to the auditorium latest by 2:15 p.m.
ये है दिन की शुरुआत हमारी पूरी एनर्जी के साथ। पर दिन खत्म होते होते हम खुद खत्म हो जाते हैं। कम ऑन गाइस, अभी दिन बाकी है। रोज लीजिए न्यूट्रेला डेली एक्टिव, जिसमें है 13 विटामिन, 12 मिनरल्स, 8 एसेंशियल अमीनो एसिड।